I welcome you all to Prof.H.Shekar Shetty Endowment Lecture Program, jointly organized by the University Endowment Lecture Organizing Scheme and the Internal Quality Assurance Cell. Today marks an important moment as we gather to acknowledge the contributions of a remarkable academician, Prof.H.Shekar Shetty. This program serves as a platform to celebrate academic excellence and intellectual discourse. It's an occasion where distinguished experts in the field of life sciences converge to share insights that inspire our academic community. ಕಾರ್ಯಕ್ರಮವನ್ನು ಪ್ರಾರ್ಥನೆಯೊಂದಿಗೆ ಪ್ರಾರಂಭಿಸ್ತಾ ಇದ್ದೀವಿ. ಮಹದೇಶ್ವರನ ಕುರಿತ ಭಕ್ತಿಗೀತೆಯನ್ನು ಈಗ ಪ್ರಸ್ತುತಪಡಿಸಲಿದ್ದಾರೆ ಡಾಕ್ಟರ್ ಲಿಂಗರಾಜು ಮತ್ತು ತಂಡದವರು.
Hey 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 hey, la 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 la, ha 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 ha, la 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 la, la 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 la. ಮೂಡಲಗಿರಿಯೋನೆ ಮುದ್ದಾದ ಮಲೆಯೋನೆ, ಬಾಳಲ್ಲಿ ಬೆಳಕ ತಂದೋನೆ, ಈ ಬಾಳಲ್ಲಿ ಬೆಳಕ ತಂದೋನೆ. ಮೂಡಲಗಿರಿಯೋನೆ ಮುದ್ದಾದ ಮಲೆಯೋನೆ, ಬಾಳಲ್ಲಿ ಬೆಳಕ ತಂದೋನೆ, ಈ ಬಾಳಲ್ಲಿ ಬೆಳಕ ತಂದೋನೆ. ನಿನ್ನಂತೆ ಬೇರೆ ನಾ ಕಾಣೆ, ಪಾದವ ಬಿಡನು ನನ್ನಾಡೆ. ನಿನ್ನಂತೆ ಬೇರೆ ನಾ ಕಾಣೆ, ಪಾದವ ಬಿಡನು ನನ್ನಾಡೆ. ಓ ಮಾದೇವ ಎಂದು ಮಾದೇವ ಎಂದು ನಾ ಕೂಗಿದಾಗ ಓಡಿ ಬಂದು ಬಾಳಲಿ ಬೆಳಕ ತಂದೋನೆ. ಮೂಡಲಗಿರಿಯೋನೆ ಮುದ್ದಾದ ಮಲೆಯೋನೆ, ಬಾಳಲ್ಲಿ ಬೆಳಕ ತಂದೋನೆ, ಈ ಬಾಳಲ್ಲಿ ಬೆಳಕ ತಂದೋನೆ. ಹೇ 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 ಹೇ, ಲಾ 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 ಲಾ, ಹಾ 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 ಹಾ, ಲಾ 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 ಲಾ, ಲಾ 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 ಲಾ. ಹೋ ಮಲಗೆ ನಾ ಬಂದೆ ಮಹದೇವಗ ಶರಣೆಂದೆ, ಮಲ್ಲಿಗೆ ಹೂ ಚೆಲ್ಲಿ ಮನಸಾರೆ ಹೊಗೆಯೆಂದೆ. ಮಲಗೆ ನಾ ಬಂದೆ ಮಹದೇವಗ ಶರಣೆಂದೆ, ಮಲ್ಲಿಗೆ ಹೂ ಚೆಲ್ಲಿ ಮನಸಾರೆ ಹೊಗೆಯೆಂದೆ. ಕಷ್ಟವಗಳ ಹೆಂದೆ ಕೈ ಹಿಡಿದು ಸಲಹೆಂದೆ, ನೆಮ್ಮದಿ ನೀಡೆಂದು ಶಿರವಾಗಿ ನಾನೆಂದೆ. ಓ ಮಾದೇವ ಎಂದು ಮಾದೇವ ಎಂದು ನಾ ಕೂಗಿದಾಗ ಓಡಿ ಬಂದು ಬಾಳಲಿ ಬೆಳಕ ತಂದೋನೆ. ಮೂಡಲಗಿರಿಯೋನೆ ಮುದ್ದಾದ ಮಲೆಯೋನೆ, ಬಾಳಲ್ಲಿ ಬೆಳಕ ತಂದೋನೆ, ಈ ಬಾಳಲ್ಲಿ ಬೆಳಕ ತಂದೋನೆ. ಹೇ 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 ಹೇ, ಲಾ 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 ಲಾ, ಹಾ 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 ಹಾ, ಲಾ 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 ಲಾ, ಲಾ 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 ಲಾ. ಕಂಬನಿ ನೀಗಿಸಿರಿ ಬೆಂಗಾವಲು ನೀನಾದೆ, ಪಾಮರ ಜನ್ಮವನು ನೀ ಪಾವನವಾಗಿಸಿರಿ. ಆ ಕಂಬನಿ ನೀಗಿಸಿದೆ ಬೆಂಗಾವಲು ನೀನಾದೆ, ಪಾಮರ ಜನ್ಮವನು ನೀ ಪಾವನವಾಗಿಸಿದೆ. ನಂಬಿದ ಭಕ್ತರನು ಕಾಪಾಡು ಎಂದು ನಿನ್ನಯ ಬಳಿ ಬಂದು ಗುಡಿ ಬಾಗಿಲಲ್ಲಿ ನಿಂದು. ಓ ಮಾದೇವ ಎಂದು ಮಾದೇವ ಎಂದು ನಾ ಕೂಗಿದಾಗ ಓಡಿ ಬಂದು ಬಾಳಲಿ ಬೆಳಕ ತಂದೋನೆ. ಶರಣು ಶರಣಯ್ಯ ನಿಮ್ಮ ಪಾದಕ ಶರಣಯ್ಯ, ಮನದೇವ ಮಲಯ ಮಾದಯ್ಯ, ಮನದೇವ ಮಲಯ ಮಾದಯ್ಯ. ಹೇ 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 ಹೇ
ಸಾದರಪಡಿಸಲಾಗುವುದು. ಸಭಾಸದರು ದಯಮಾಡಿ ನಾಡಗೀತೆಗೆ ಗೌರವ ಸೂಚಕವಾಗಿ ಎದ್ದು ನಿಲ್ಲಬೇಕಾಗಿ ವಿನಂತಿಸುತ್ತೇನೆ.
जय भारत जननीय तनुजाते, जय हे कर्नाटक माते, जय सुंदर नदीवनಗಳ ನಾಡೆ, जय हे ರಸ ಋಷಿಗಳ ಬೇರೆ. ಭೂದೇವಿಯ ಮಕುಟದ ನವಮಣಿಯೇ, ಗಂಧದ ಚಂದದ ಹೊನ್ನಿನ ಗಣಿಯೇ, ರಾಘವ ಮಧುಸೂದನರವತರಿಸಿ ಭಾರತ ಜನನಿಯ ತನುಜಾತೆ. ಜನನಿಯ ಜೋಗುಳ ವೇದದ ಘೋಷ, ಜನನಿಗೆ ಜೀವವು ನಿನ್ನಾವೇಶ. ಹಸುರಿನ ಗಿರಿಗಳ ಸಾಲೆ, ನಿನ್ನಯ ಕೊರಳಿನ ಮಾಲೆ. ಕಪಿಲ ಪತಂಜಲ ಗೌತಮ ಜಿನನು ಭಾರತ ಜನನಿಯ ತನುಜಾತೆ. ಶಂಕರರ ಮನುಜ ವಿದ್ಯಾರಣ್ಯ ಬಸವೇಶ್ವರ ಮಧ್ವ ದಿವ್ಯಾರಣ್ಯ. ರಂಗ ಷಡಕ್ಷರಿ ಪೊನ್ನ ಪಂಪಲ ಕುಮಿಪತಿ ಜನ್ನ. ಕುಮಾರ ವ್ಯಾಸರ ಮಂಗಳ ಧಾಮ, ಕೋಗಿಲೆಗಳ ಪುಣ್ಯಾರಾಮ. ಆನಕ ರಾಮಾನಂದ ಕಬೀರ ಭಾರತ ಜನನಿಯ ತನುಜಾತೆ. ತೈಲ ಪವೈತಳರಾಳಿದ ನಾಡೆ, ಕಂಕಣ ಜಗನರ ನೆಚ್ಚಿನ ಬೀಡೆ. ಕೃಷ್ಣ ಶರಾವತಿ ತುಂಗ, ಕಾವೇರಿಯವರ ರಂಗ. ಚೈತನ್ಯ ಪರಮಹಂಸ ವಿವೇಕ ಭಾರತ ಜನನಿಯ ತನುಜಾತೆ. ಸರ್ವ ಜನಾಂಗದ ಶಾಂತಿಯ ತೋಟ, ರಸಿಕರ ಕಂಗಳ ಸೆಳೆಯುವ ನೋಟ. ಹಿಂದೂ ಸೈತ ಮುಸಲ್ಮಾನ, ಪಾರಸಿಕ ಜೈನ ಉದ್ಯಾನ. ಜನಕನ ಹೋಲುವ ದೊರೆಗಳ ಧಾಮ, ಕಾಯಕ ವೈಣಿಕರ ರಾಮ. ಕನ್ನಡ ನುಡಿ ಕುಣಿದಾಡುವ ಗೇಹ, ಕನ್ನಡ ತಾಯಿಯ ಮಕ್ಕಳ ದೇಹ. ಭಾರತ ಜನನಿಯ ತನುಜಾತೆ, ಜಯ ಹೇ ಕರ್ನಾಟಕ ಮಾತೆ. ಜಯ ಹೇ ಕರ್ನಾಟಕ ಮಾತೆ, ಜಯ ಹೇ ಕರ್ನಾಟಕ ಮಾತೆ. ಧನ್ಯವಾದಗಳು.
ಪ್ರಾರ್ಥನೆ ಹಾಗೂ ನಾಡಗೀತೆಯನ್ನು ಹಾಡಿದ ಡಾಕ್ಟರ್ ನಿಂಗರಾಜು, ಡಾಕ್ಟರ್ ಮಧುಸೂದನ್ ಹಾಗೂ ಡಾಕ್ಟರ್ ಲೋಕೇಶ್ ಅವರಿಗೆ ತಮ್ಮೆಲ್ಲರ ಪರವಾಗಿ ವಂದನೆಗಳನ್ನ ಸಲ್ಲಿಸ್ತೇನೆ. Today, we have amongst with us Dr. K. Amritesh, who is serving as a professor at the Department of Studies in Botany. Recently, he has been honored with the National Academy of Biological Sciences Fellowship for his remarkable contributions to agricultural sciences. Dr. Amritesh holds a distinguished academic record, including a doctoral degree from the University of Mysore and a postdoctoral degree from the University of North Texas. His pioneering research in plant microbe interactions and nanotechnology has resulted in 130 publications with an impressive H index of 34 and over 3,600 citations, marking him as a trailblazer in his field. His leadership as director of the Internal Quality Assurance Cell of the university underscores his pivotal role in academia. As an ardent student of Prof. Shetty, Dr. Amritesh embodies his mentor's value of scholarly pursuit. Additionally, he serves as the coordinator of the University Endowment Lecture Organizing Scheme, which hosts this program. It is now my honor to invite Dr. Amritesh to extend a warm welcome to our esteemed guests and invitees to this significant event.
ಎಲ್ಲರಿಗೂ ನನ್ನ ನಮಸ್ಕಾರಗಳು. Honorable Vice Chancellor Professor N.K. Loknath, today's Chief Guest Professor S.R. Niranjana, Vice Chairman Karnataka State Higher Education Council. Another Chief Guest of the day, Professor Apparao Kodale, former Vice Chancellor Central University of Hyderabad and also J.C. Bose Fellow, distinguished fellow, and who is the fellow of all the prestigious academy of the nation. Mrs. V.R. Shailaja, Registrar, University of Mysore, my mentor, Professor H.Shekar Shetty, and invitees, Professor S. Shankar Bhatt, Professor H.S. Prakash, Dr. K. Madhav, Syndicate Member, and senior faculty, Professor J. Janardana, Professor Kempraj, Professor M.I. Srinivasa, donor, Dr. G. Chalvaraju, and my own learned faculty members, fellow colleagues, Dr. S. Basurajappa, Dr. G. Manjunath, Dr. M.S. Sharda, Dr. M.S. Nalini, and other learned colleagues, and importantly, the interest shown by our campus students, all the 9 life sciences students who have gathered to overwhelming response to our endowment lecture. Really, we are very honored, and our all chief guests are very happy by seeing you all in one umbrella. Thank you very much, first of all, to you and media personnel. And I would also welcome Dr. J. Lohit, who is also the organizing committee coordinator of this program, who is also here, and my IQAC staff and Department of Botany office staff. So, first of all, the purpose of holding the university endowment lecture program, especially in the name of Prof.H.Shekar Shetty, who has contributed significantly for the establishment of Department of Applied Botany and Biotechnology. His contribution in the area of especially the plant disease diagnosis and host-plant interaction and eco-friendly management of diseases have resulted attraction to the various companies around the globe, especially the private companies who are the beneficiaries of his research. When he has undertaken as a postdoctoral fellow at the Danish Government Institute of Seed Pedagogy, DJSP, Denmark, Copenhagen. So where he has conducted for the first time pulmonary downy mildew screening procedure, and it was published in 1980 as an embryo count method that has resulted a great turning point for the establishment of Department of Applied Botany and Biotechnology on the campus, one of the leading departments on the campus, which was emerged from Department of Botany, that we cannot forget. So that is the one thing. They were working in the Department of Botany, and for the establishment of a separate laboratory called Pulmonary Downy Mildew Research Laboratory, even today funded by one of the national funding agencies called ICR, Indian Council of Agriculture Research. Because of extensive investigations on the host-plant interaction, he has produced more than 50 PhD students, more than 500 MPhil students, more than 1,000 students have been generated by the Department of Applied Botany. That is the greatest achievement, that is the greater. So, because of the, especially the quick diagnosis, molecular diagnosis and all those have fetched some of the private companies to export the seed materials. So that is why it was attracted, his research really attracted, 400 publications attracted many seed companies. But Pioneer Seed Company has come forward to institute an endowment lecture in his name. That is why we are all here. First of all, we sincerely, University of Mysore and Department of Applied Botany and Biotechnology and Botany, we sincerely, wholeheartedly thank the Pioneer Seed Company Private Limited Hyderabad for hosting this program every year. So that is why we are here. So the endowment lectures are witnessing many events on the campus. When you go back to the history, Princess Leelavati endowment lecture, Sir M. Vishweswaraya chair. So many chairs have been instituted by many donors. Professor Paul Neergard, another, and Professor Vikos Medagard, they visited the Department of Applied Botany and Biotechnology because of the chair, Sir M. Vishweswaraya chair. Otherwise, the Applied Botany and Biotechnology, whatever the name it has earned to today, it wouldn't have possible. That is the reason we have to always respect the donors and as well as the endowment lectures. That's why we are all together to hear all these developments. So all these years, Professor S.R. Niranjana, who is here now, he was working as a chairperson of the department. He has conducted several endowment lectures of the department, Professor Shekar Endowment lecture, by inviting eminent scholars of the nation, Professor Anupam Verma, Professor H.A. Ranganath, Professor P. Balram, and today another feather in the cap, Professor Apparao Padale, FNA, who is also delivering Professor Shekar Shetty. It's an honor to the university. And Professor K.S. Rangappa, then former Vice Chancellor, who has also invited Nobel laureates, more than 8 Nobel laureates. That is the another greatness during the university centenary celebration. Really great, remarkable days we had due to endowment lecture. That is why our Honorable Vice Chancellor has given me the responsibility to continuously monitor these endowment lectures that are there in our university, to update the current and contemporary issues for the benefit of scholars and faculty with the current knowledge. That's why we have been, all of us, witnessing this important moment. So with this background, I would like to welcome Honorable Vice Chancellor Professor N.K. Loknath, who is an academician. So really he's been working hard because of as soon as he assumed the office of the Honorable Vice Chancellor's position, he started conducting the academic review meeting on a weekly basis, inviting every department, meeting one-on-one faculty members and to take the stock of the situation to take up for the 4 years of his term. Really, and we have all conducted all the 55 departments and everyone knows how he is doing the things. And our Professor Mentor, Professor S.R. Niranjana, when he assumed the charges of Vice Chairman of the Karnataka State Higher Education Council, he has made a plan to introduce all over the state, whoever they are good in their subject of expert like arts, commerce, they wanted to deliver the lectures online. The first person to deliver the lecture is our Honorable Vice Chancellor and in the online platform that was introduced by the Professor S.R. Niranjana. Our Honorable Vice Chancellor came forward to deliver each area, physics, to all over the Karnataka. This is what the plan of Higher Education Council, whoever they are doing best job, they wanted to reach even to Chamarajanagar, Mandya or Belgaum, any university for that matter, if they are good. So that's why that was initiated by Higher Education Council. The first person to come forward is our Honorable Vice Chancellor. First lecture given by our Honorable Vice Chancellor for all the Karnataka postgraduate students of physics. So that is the greatness. And even today, he takes every day the classes. He comes to physics department. He has become a model, trendsetter for all the. He knows who are all taking the classes, who are all not taking the classes. He knows, but he will take some other platform to what is going on, the performance of the faculty members and students. He comes and he visits all the 55 departments and he knows the stock of the situation of the campus, how he has to develop. He has his own plans. So research on research also, he's been recently given top-cited scientist. So that is also a great credit of our Honorable Vice Chancellor, who is here, and on behalf of University Endowment Lecture and on behalf of IQAC, we wholeheartedly welcome you, sir, for this very important, significant this one. I request Professor G.R. Janardana to welcome our Honorable Vice Chancellor to this function. Another important chief guest and my mentor, Professor S.R. Niranjana, who has occupied the highest position of being an alumnus of University of Mysore, is a great pride for all of us because he is the head for all the 34 state universities and after serving as Gulbarga University Vice Chancellorship. So as soon as he has completed the Vice Chancellorship, still he was left with 2 years of service. Then Vice Chancellor Professor G. Hemant Kumar, no, you are not supposed to take rest. You should come as a chairperson of the department. That has another remarkable change for the Department of Applied and Biotechnology because he has constructed a new wing and he has made wonderful laboratory the way Prof.H.Shekar Shetty has made already. So he has added the same to the same good old building and he has served the department for 2 years, brought the discipline and in all the respect, made all the facilities for the department actually. So that is another credit. So in Gulbarga University, he made remarkable reforms by bringing, so especially the North Karnataka, the dry weather, but in spite of it, he could planted more than 10,000 saplings on the campus, made greenery. So all of a sudden, so because of under 4 years regime, that is also his greatness, I can say. So with regard to academic achievements, he has done a wonderful job. So especially the coffee growers. So he has come out the solution actually. The problem faced by the coffee growers, especially Bavaria Bassiana. So that was the major problem. So at that time he was young when he started his career in 1992. He started working with Prof.H.Shekar Shetty under the guidance of Professor Shetty, come out with the solution of Bavaria Bassiana, an entomopathogenic fungus, and spread the solution and became a challenge to coffee board when he could not able to control that particular diseases. that has brought him laurels from DBT, and from then onwards he started his career in a wonderful way. He has come out very well, and he has also been recognized various ways. Karnataka State Academy and all the 3 academies, Fellow of Indian National Academy of Sciences, Fellow of Allahabad Academy, and all those academy that has given fellowship to him, especially Dr. Rajaramanna, Dr. Sarameshwara awards, and the Sir C.V. Raman Young Scientist Award brought him for these academic achievements. Really very honored being our own mentor and being the alumnus of our campus, Department of Botany and as well as the Department of Biotechnology. So he is serving for the good cause, and he is helping us, our university, with our Honorable Vice Chancellor, interacting with our Registrar and Honorable Vice Chancellor for the development of this university, his own university. That's what we were discussing, and on behalf of University Endowment Lecture and on behalf of IQAC, I wholeheartedly welcome, I request Dr. M.Y. Srinivasa to welcome Professor S.R. Niranjana on this occasion. So today's chief guest, a distinguished fellow of the nation, the Central University of Hyderabad, which is the national university of national importance, who has served the Vice Chancellor of Central University, Professor Apparao Podile, who is also a distinguished fellow. All the academy, 3 academy, distinct FNA, FNAC, FNA, all the 3 fellows of the nation. So who is here, who is going to deliver the area of Professor Shekar Shetty, Dr. S.R. Niranjana, Dr. Prakash, and all those areas of the similar nature. They are working on osmo interaction, especially the chitin oligosaccharides, the need of the hour. So that he is going to deliver, who is also JC Fellow, who is here for the delivering a special lecture on the occasion of Professor Shekar Shetty Endowment Lecture Program. The details will be given by Professor Dr. J. Lohit in the later. So I wholeheartedly welcome Dr. Apparao Podile. I request Dr. N.S. Raju, Chairperson Environmental Science, to welcome Professor Apparao Podile on this occasion. Please welcome, sir. Please. Thank you very much. Now another important personality on the dais, Mrs. V.R. Shailaja, who is an administrator, very effectively running the campus, very effective manner, who has served in various capacities in the state government, who is a senior KS officer and alumna of Department of Studies in Geography, who is been here and helpful for all the academic program, and she is also here, and I welcome Mrs. V.R. Shailaja on this occasion, please. Thank you, madam. So the way we are all gathered to mark the significant role of as a model to the campus trendsetter, Professor Shekar Shetty, who is here, who has brought 3 important wings. One is mycology, virology and bacteriology. 3 wings like a ಭೂಸೇನೆ, ವಾಯುಸೇನೆ ತರ ನಮ್ಮ ಡಾಕ್ಟರ್ ಶೇಖರ್ ಶೆಟ್ಟಿ ಅವರು, ಪ್ರೊಫೆಸರ್ H.S. ಪ್ರಕಾಶ್, ಪ್ರೊಫೆಸರ್ S.R. ನಿರಂಜನ, ಪ್ರೊಫೆಸರ್ ಎಂ ಕೃಷ್ಣಪ್ಪ ಅವ್ರನ್ನ ಬೆಳೆಸಿದಂಥ ಈ ಒಂದು ಸಂದರ್ಭದಲ್ಲಿ ಈ 3 ವಿಂಗು ಇವತ್ತು ದೇಶಾದ್ಯಂತ, ರಾಷ್ಟ್ರಾದ್ಯಂತ, ವಿಶ್ವದಾದ್ಯಂತ ತನ್ನದೇ ಆದಂಥ ಖ್ಯಾತಿಯನ್ನು ಪಡೆದಿದ್ದಾರೆ. 3 ಪ್ರೊಫೆಸರ್ಗಳನ್ನ ನಡುವೆ ಅವರು ಬೆಳೆಸಿದಂಥ ವಿಶ್ವವಿದ್ಯಾನಿಲಯ ನಿಜವಾಗ್ಲೂ ಅಂತಾರಾಷ್ಟ್ರೀಯ ಮಟ್ಟಕ್ಕೆ ಹೋಗಿದೆ. ಅವ್ರಿಗೂ ಸಹ ಈ ಒಂದು ಸಂದರ್ಭದಲ್ಲಿ ತುಂಬ ಆತ್ಮೀಯವಾಗಿ ತಮ್ಮನ್ನು ಬರಮಾಡ್ಕೊಳ್ತಾ ಇದ್ದಾರೆ ಸರ್. ಥ್ಯಾಂಕ್ ಯು, ವೆಲ್ಕಮ್ ಸರ್. ಥ್ಯಾಂಕ್ ಯು. ಡಾಕ್ಟರ್ ಜಿ. Welcome all the faculty members, various department chairpersons, especially we have invited Department of Biotechnology, Botany and Microbiology and Environmental Science, Biochemistry, Molecular Biology, Zoology, Sericulture Science, Genetics and Genomics, and especially the MSc Botany students from JSS Botany and Marnis Botany and St. Philomena's Botany students are here. We are really very happy for your visit. So you are going to hear a wonderful lecture that is arranged under the banner of University of Mysore Endowment Lecture Series, and all the faculty members, and especially in welcoming Dr. K. Mahadev, ex-farmer syndicate member, who is also here witnessing my mentor's close friend and Dr. S.R. Niranjana's close friend. So who is here, Dr. T. Vasant Kumar, Dr. H.S. Prakash, Dr. Shankar, but who worked closely with the Professor Shekar Shetty, who is also here, and all my fellow colleagues, media personnel and students. So I welcome all of you for this significant endowment lecture. Thank you very much. Once again, all of welcome everyone. Thank you very much.
ಎಲ್ಲರನ್ನ ಆತ್ಮೀಯವಾಗಿ ಸ್ವಾಗತಿಸಿದಂತಹ ಪ್ರೊಫೆಸರ್ ಅಮೃತೇಶ್ ಅವರಿಗೂ ಕೂಡ ತಮ್ಮೆಲ್ಲರ ಪರವಾಗಿ ಹೃತ್ಪೂರ್ವಕವಾದ ಸ್ವಾಗತವನ್ನು ಬಯಸ್ತೇನೆ. At its heart, this endowment lecture program aims to uphold the values of scholarship, research and innovation that Professor Shekar Shetty embodied throughout his illustrious career. It is now my privilege to invite Professor S.R. Niranjana, a distinguished academic leader and a long-time associate of Professor Shetty, to inaugurate this year's Shekar Shetty Endowment Lecture Program by lighting the ceremonial lamp. I also invite other dignitaries present on the dais to join hands with Professor Niranjana in this. ಕಾರ್ಯಕ್ರಮವನ್ನು ವಿದ್ಯುಕ್ತವಾಗಿ ಉದ್ಘಾಟಿಸಿದ ಗಣ್ಯರೆಲ್ಲರಿಗೂ ವಂದನೆಗಳನ್ನ ಸಲ್ಲಿಸ್ತೀನಿ. Ladies and gentlemen, it is my great pleasure to introduce Professor S.R. Niranjana, a true luminary in the field of higher education. In a remarkable 35-year career, Professor Niranjana has guided 21 students for their doctoral degree, published over 213 research articles with an H-index of 42 and over 5,600 citations to his credit. He has successfully organized more than 25 national and international conferences. Notably, he held key positions such as the Vice Chancellor of Gulbarga University. Currently, he is the Vice Chairman of Karnataka State Higher Education Council and also the lifelong distinguished professor and UGC DSR faculty fellow at the University of Mysore. Under his leadership, the Institute of Eminence at the University of Mysore was established, which got funding from the MHRD. He superintended a European Union program for seed treatment strategies in medicinal plants in India and Africa. Professor Niranjana has been honored with prestigious titles from distinguished academies. He is a fellow of the National Academy of Sciences, the National Academy of Agricultural Sciences, and the National Academy of Biological Sciences. Internationally, he is recognized as a fellow of the Danish International Development Agency, Denmark. Awardee of numerous accolades, including Sir Visheshwaraya Award, Sir C.V. Raman Birth Centenary Award, Dr. Raja Ramanna State Award, Professor S.S. Chahal Lifetime Achievement Award, and Maharshi Valmiki Award, Professor S.R. Niranjana stands as a distinguished scholar, leaving an enduring impact on the field of plant pathology and development. Now, I kindly request Professor Niranjana to deliver the inaugural address, setting the stage for an enlightening and thought-provoking discussion.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ಮೈಸೂರು ವಿಶ್ವವಿದ್ಯಾಲಯ ಹಾಗೂ ಇಂಟರ್ನಲ್ ಕ್ವಾಲಿಟಿ ಅಶ್ಯೂರೆನ್ಸ್ ಸೆಲ್ ಸಂಯುಕ್ತಾಶ್ರಯದಲ್ಲಿ ಪ್ರೊಫೆಸರ್ ಶೇಖರ್ ಶೆಟ್ಟಿ ಎಂಡೋನ್ಮೆಂಟ್ ಲೆಕ್ಚರನ್ನು ಆಯೋಜಿಸತಕ್ಕಂಥ ಸಂದರ್ಭದಲ್ಲಿ ವೇದಿಕೆ ಮೇಲೆ ಆಸೀನರಿರ್ತಕ್ಕಂಥ ಮೈಸೂರು ವಿಶ್ವವಿದ್ಯಾಲಯದ ಗೌರವಾನ್ವಿತ ಕುಲಪತಿಗಳಾದಂಥ ಸ್ನೇಹಿತರಾದಂಥ ಪ್ರೊಫೆಸರ್ ಎನ್.ಕೆ. ಲೋಕನಾಥ್ ಅವರೇ, ಪ್ರೊಫೆಸರ್ ಶೇಖರ್ ಶೆಟ್ಟಿ ಎಂಡೋನ್ಮೆಂಟ್ ಲೆಕ್ಚರ್ನ ಅಂಗವಾಗಿ ಉಪನ್ಯಾಸವನ್ನು ನೀಡಲು ಆಗಮಿಸಿರ್ತಕ್ಕಂಥ ಆತ್ಮೀಯ ಸ್ನೇಹಿತರಾದಂಥ ಮತ್ತು ಹೈದರಾಬಾದ್ ಸೆಂಟ್ರಲ್ ಯೂನಿವರ್ಸಿಟಿಯ ಫಾರ್ಮರ್ ವೈಸ್ ಚಾನ್ಸಲರ್ ಆದಂಥ ಪ್ರೊಫೆಸರ್ ಅಪ್ಪುರಾವ್ ಪ್ರಳಯ ಅವರೇ, ಗುರುಗಳು ಮತ್ತು ಹಿತೈಷಿಗಳು ಆದ ನಮ್ಮೆಲ್ಲರ ನೆಚ್ಚಿನ ಗುರುಗಳಾದಂಥ ಪ್ರೊಫೆಸರ್ H. ಶೇಖರ್ ಶೆಟ್ಟಿ ಅವರೇ, ಈಗ ತಾನೇ ಅವರ ತುರ್ತು ಕೆಲಸದ ಅಂಗವಾಗಿ ನಿರ್ಗಮಿಸಿ ಹೋದಂಥ ಮೈಸೂರು ವಿಶ್ವವಿದ್ಯಾಲಯದ ಕುಲಸಚಿವರಾದ ಶ್ರೀಮತಿ V.R. ಶೈಲಾಜರವರೇ, ಇದಕ್ಕೆ ಮುಂದೆ ಆಸೀನರಾಗಿರ್ತಕ್ಕಂಥ ನಮ್ಮೆಲ್ಲರ ಹಿತೈಷಿಗಳಾದ ಮತ್ತು ಮೈಸೂರು ವಿಶ್ವವಿದ್ಯಾಲಯದ ಮಾಜಿ ಸಿಂಡಿಕೇಟ್ ಸದಸ್ಯರಾದಂಥ ಡಾಕ್ಟರ್ ಕೆ ಮಾದೇವರವರೇ, ಗುರುಗಳಾದಂಥ ಪ್ರೊಫೆಸರ್ ಶಂಕರ್ ಭಟ್ರವರೇ, ಪ್ರೊಫೆಸರ್ ಪ್ರಕಾಶ್ ರವರೇ, ಡಾಕ್ಟರ್ ವಸಂತ್ ಕುಮಾರ್ ಅವರೇ ಮತ್ತು ಎಲ್ಲ ವಿಭಾಗಗಳ ಮುಖ್ಯಸ್ಥರುಗಳೇ, ಇಲ್ಲಿ ಆಗಮಿಸಿರ್ತಕ್ಕಂಥ ಎಲ್ಲ ವಿದ್ಯಾರ್ಥಿಗಳೇ, ಮಾಧ್ಯಮ ಮಿತ್ರರೇ ಮತ್ತು ಎಲ್ಲ ಸ್ನೇಹಿತರೇ, ಸಂಶೋಧಕರು. ಈಗ ತಾನೇ ಕೆ ಎನ್ ಅಮೃತೇಶ್ ಅವರು ಬಹಳ ನಿರಾರ್ಗಳವಾಗಿ ಈ ಉಪನ್ಯಾಸದ ಒಂದು ಪ್ರಾಮುಖ್ಯತೆಯನ್ನು ತಮಗೆಲ್ಲ ತಿಳಿಸ್ಕೊಟ್ಟಿದ್ದಾರೆ. ಹಾಗಾಗಿ ಈ ದತ್ತಿ ಉಪನ್ಯಾಸ ನಡೆದು ಬಂದ ದಾರಿಯ ಬಗ್ಗೆ ನಾನು ಹೆಚ್ಚು ಇದನ್ನು ಮಾತಾಡಲಿಕ್ಕೆ ಹೋಗುವುದಿಲ್ಲ. ಎಷ್ಟು ದತ್ತಿ ಉಪನ್ಯಾಸಗಳು ಮೈಸೂರು ವಿಶ್ವವಿದ್ಯಾಲಯ ಇದ್ದಾವೆ, ಅಷ್ಟು ದತ್ತಿ ಉಪನ್ಯಾಸಗಳು ಪ್ರತಿ ವರ್ಷ ನಡೀಬೇಕು ಅಂತಕ್ಕಂಥದ್ದು ಕುಲಪತಿಗಳಲ್ಲಿ ನನ್ನದೊಂದು ಮನವಿ. ಎಲ್ಲ ದತ್ತಿ ಉಪನ್ಯಾಸಗಳನ್ನ ಅವರವರ ಹೆಸರುಗಳಲ್ಲಿ ಇಲ್ಲಿ ಏನು ಅದನ್ನ ಇನ್ಸ್ಟಿಟ್ಯೂಟ್ ಮಾಡಿದ್ದಾರೆ, ಅದರ ಗೌರವಾತ್ಮಕವಾಗಿ ಮಾಡಿರೋದ್ರಿಂದ ವಿಶ್ವವಿದ್ಯಾಲಯ ಅದನ್ನ ಗಂಭೀರವಾಗಿ ಪರಿಗಣಿಸಿ ಬರೋ ದಿನಗಳಲ್ಲಿ ಎಲ್ಲ ಎಂಡೋನ್ಮೆಂಟ್ಗಳು, ನಾಟ್ ಓನ್ಲಿ ಶೇಖರ್ ಶೆಟ್ಟಿ ಎಂಡೋನ್ಮೆಂಟ್ ಅಲ್ಲಿ ಯಾವುದೇ ಎಂಡೋನ್ಮೆಂಟ್ ಇದ್ರೂ ಸಹ ಅದನ್ನ ಪ್ರತಿ ವರ್ಷ ನಡೀತಕ್ಕಂಥ ಒಂದು ಕೆಲಸವನ್ನ ಕೈಗೊಳ್ಳಬೇಕು ಅಂತ ಹೇಳಿ ನಾನು ಮೈಸೂರು ವಿಶ್ವವಿದ್ಯಾಲಯದ ಕುಲಪತಿಯಲ್ಲಿ ಮನವಿಯನ್ನು ಮಾಡ್ತೀನಿ. ಪ್ರೊಫೆಸರ್ ಶೇಖರ್ ಶೆಟ್ಟಿ ಅವ್ರ ಬಗ್ಗೆ ಈಗಾಗಲೇ ಅಮೃತೇಶ್ ಅವರು ಬಹಳ ವಿಚಾರಗಳನ್ನು ತಮಗೆ ಹೇಳಿದ್ದಾರೆ. ನಾನು ಅವರ ವಿದ್ಯಾರ್ಥಿಯಾಗಿ ಮತ್ತು ಅವರ ಸಹೋದ್ಯೋಗಿಯಾಗಿ ಎಲ್ಲ ಅಂಗಗಳಲ್ಲಿ ಕೆಲಸ ಮಾಡಿರ್ತಕ್ಕಂಥ ಸಂದರ್ಭದಲ್ಲಿ ನಾವು ವಿಶ್ವವಿದ್ಯಾಲಯದಲ್ಲಿ ಕಂಡಂತ ಒಬ್ಬ ಬಹಳ ಬಹಳ ಗೌರವಾನ್ವಿತ ಪ್ರಾಧ್ಯಾಪಕರು ಅಂತ ಹೇಳಲಿಕ್ಕೆ ಈ ಸಂದರ್ಭದಲ್ಲಿ ನಾನು ಇಷ್ಟಪಡ್ತೇನೆ. ಹಾಗಾಗಿ ಅವರ ಹೆಸರಲ್ಲಿ ಒಂದು DuPont ಅಂತಕ್ಕಂಥ ಒಂದು ಸೀಡ್ ಕಂಪನಿ ಈ ಎಂಡೋನ್ಮೆಂಟ್ ಅನ್ನ ಇನ್ಸ್ಟಿಟ್ಯೂಟ್ ಬಹಳ ಹಿಂದೆ ಮಾಡಿದ ಸಂದರ್ಭದಲ್ಲಿ ಪ್ರೊಫೆಸರ್ ಶೇಖರ್ ಶೆಟ್ಟಿ ಅವರು 2009ರಲ್ಲಿ ನನ್ನನ್ನು ಕೋಆರ್ಡಿನೇಟರ್ ಆಗಿ ಮಾಡಿದರು. ನಾನು 2015ರವರೆಗೆ ಆ ಎಂಡೋನ್ಮೆಂಟ್ ಅನ್ನ ಮಾಡ್ಕೊಂಡು ಬಂದೆ. ಈಗಾಗಲೇ ಅಮೃತೇಶ್ ಅವರು ಹೇಳಿದಾಗೆ ಎಲ್ಲ ಸ್ಕಾಲರ್ಗಳನ್ನ ಪ್ರೊಫೆಸರ್ ಶೇಖರ್ ಶೆಟ್ಟಿ ಅವರ ಒಂದು ಸೈಂಟಿಫಿಕ್ ಕಾಂಟ್ರಿಬ್ಯೂಷನ್ ಈಸ್ ರೆಕಗ್ನೈಸ್ಡ್ ಓವರ್ ದಿ ವರ್ಲ್ಡ್. ನಾನು ಆಗಲೂ ಅಮೃತೇಶ್ ಹೇಳಿದೆ, 1 ವರ್ಷದಲ್ಲಿ ಡಾಕ್ಟರ್ S.B. ಮಾಥುರವನ್ನು ಸಹ ಎಂಡೋನ್ಮೆಂಟ್ಗೆ ಕರಿಬೇಕು ಅಂತಕ್ಕಂಥದ್ದಲ್ಲಿ ನಾವು ಇವತ್ತು ಈ ಡಿಪಾರ್ಟ್ಮೆಂಟ್ ಬೆಳಿಲಿಕ್ಕೆ, ನಾವೆಲ್ಲ ಅದರ ಆಶ್ರಯದಲ್ಲಿ ಬೆಳಿಲಿಕ್ಕೆ ಆ ಡೆನಿಡಾ ಅಂತ ಒಂದು ಪ್ರೋಗ್ರಾಮು ನಮ್ಮನ್ನು ಬಹಳ ಎತ್ತರಕ್ಕೆ ತಗೊಂಡು ಹೋಗುವಂತ ಸಂದರ್ಭ. ಎಲ್ಲ ವಿದ್ಯಾರ್ಥಿಗಳು, ಅಧ್ಯಾಪಕರು ನಾವೆಲ್ಲ ಅಲ್ಲಿ ಒಂದು ಟ್ರೈನಿಂಗ್ ಅನ್ನು ತಗೊಂಡು ಆ ಡೆನಿಡಾ ಅಂತಕ್ಕಂಥ ಒಂದು ಸಂಸ್ಥೆ ನಮ್ಮ ಸಂಸ್ಥೆಗೆ ಬಹಳ ಒತ್ತನ್ನು ಕೊಟ್ಟು ನಾವೆಲ್ಲ ಬಹಳ ಮೇಲಕ್ಕೆ ಬೆಳೀತಕ್ಕಂಥ ಒಂದು ಅವಕಾಶವನ್ನು ಮಾಡಿಕೊಟ್ಟಿದ್ದರಿಂದ ಡಾಕ್ಟರ್ S.B. ಮಾಥುರ್ ಅವರನ್ನ ಒಮ್ಮೆ ಈ ಎಂಡೋನ್ಮೆಂಟ್ ಲೆಕ್ಚರ್ಗೆ ವಿಶ್ವವಿದ್ಯಾಲಯ ಕರಿಬೇಕು. ಇದು ಇನ್ನು ಮುಂದೆ ವಿಶ್ವವಿದ್ಯಾಲಯದ ಕಾರ್ಯಕ್ರಮ. ಯಾವುದೇ ಎಂಡೋನ್ಮೆಂಟ್ ವಿಶ್ವವಿದ್ಯಾಲಯದಲ್ಲಿದ್ದಾಗ ಯಾವುದೇ ವಿಭಾಗದ ಕಾರ್ಯಕ್ರಮ ಇದು ಆಗುವುದಿಲ್ಲ. ಇದು ವಿಶ್ವವಿದ್ಯಾಲಯದ ಕಾರ್ಯಕ್ರಮ. ರಿಜಿಸ್ಟ್ರಾರು ಮತ್ತು ಕುಲಪತಿಗಳು ಎಂಡೋನ್ಮೆಂಟ್ಗಳನ್ನ ಜವಾಬ್ದಾರಿಯಿಂದ ನಡೆಸ್ಕೊಂಡು ಹೋಗ್ತಕ್ಕಂಥ ಒಂದು ಸಂದರ್ಭ ನಿರ್ಮಾಣ ಆಗಬೇಕು ಅಂತಕ್ಕಂಥದ್ದು ನನ್ನ ಮನವಿ. ನಾನು ಗುಲ್ಬರ್ಗದಲ್ಲಿ ಕುಲಪತಿ ಆಗಿದ್ದಾಗ ಹಾಗೆ ಮಾಡಿ 27 ಚೇರ್ಗಳಿದೆ. ಗುಲ್ಬರ್ಗದಲ್ಲಿ ಇರ್ತಕ್ಕಂಥಷ್ಟು ಚೇರ್ಗಳು ಎಂಡೋನ್ಮೆಂಟ್ಗಳು ಬಹುಶಃ ಕರ್ನಾಟಕದ ಯಾವ ವಿಶ್ವವಿದ್ಯಾಲಯದಲ್ಲೂ ಸಹ ಇಲ್ಲ. 104 ಎಂಡೋನ್ಮೆಂಟ್, 27 ಚೇರ್ಗಳು ಸಾಲದ್ದಕ್ಕೆ ಪ್ರೊಫೆಸರ್ ಅಮಾನಾಯಕರು ಮಾಡಿಟ್ಟು ಬಂದಂತ ಒಂದು ರಾಜ್ಯೋತ್ಸವ, ಕರ್ನಾಟಕ ರಾಜ್ಯೋತ್ಸವ ಪ್ರಶಸ್ತಿ ಅಂತಕ್ಕಂಥದ್ದು. ಆ ಭಾಗದಲ್ಲಿ ಕನ್ನಡ ಪುಸ್ತಕ ಬರೆದವರಿಗೆ ಅವಾರ್ಡ್ ಅನ್ನು ಕೊಡಬೇಕು ಅಂತೇಳಿ ಅಮಾನಾಯಕರು ಆ ವಿಶ್ವವಿದ್ಯಾಲಯದಲ್ಲಿ ಒಂದು ದತ್ತಿಯನ್ನ ಶುರು ಮಾಡಿ ಬಂದ್ರು. ಇವತ್ತು ಸಹ ನಡ್ಕೊಂಡು ಬಂದಿದೆ. ಆ ಭಾಗದಲ್ಲಿ ಬರೆದಂತ ಪುಸ್ತಕಗಳಿಗೆ ಕನ್ನಡದಲ್ಲಿ, ವಿಜ್ಞಾನದಲ್ಲಿ ಬರಲಿ, ಸಾಹಿತ್ಯದಲ್ಲಿ ಬರಲಿ, ನೀವು ಕಾಮರ್ಸ್ ಅಲ್ಲಿ ಬರೀರಿ, ಯಾವುದ್ರಲ್ಲಿ ಬರೆದ್ರು ಸಹ ಅದನ್ನ ಅಲ್ಲಿ ಪುರಸ್ಕರಿಸಿ ಒಂದು ಅದಕ್ಕೆ ಅವಾರ್ಡ್ ಕೊಡ್ತಕ್ಕಂತ ಒಂದು ವಾತಾವರಣವನ್ನ ಅಮ್ಮನಾಯಕರು ಆ ವಿದ್ಯೆಯಲ್ಲಿ ಮಾಡಿ ಬಂದರು. ಇದು ಯಾಕೆ ಹೇಳ್ತೀನಿ ನಾನು ಅಂದ್ರೆ, ವೈಯಕ್ತಿಕವಾಗಿ ವ್ಯಕ್ತಿಗಳ ಗೌರವಕ್ಕಾಗಿ ಮಾಡಿರತಕ್ಕಂಥ ಕಾರ್ಯಕ್ರಮಗಳಲ್ಲ ಇವು. ವಿಶ್ವವಿದ್ಯಾಲಯವನ್ನು ಗುರುತಿಸಿ ಇದೊಂದು ವಿಶ್ವವಿದ್ಯಾಲಯದಲ್ಲಿ ನನ್ನದೊಂದು ದತ್ತಿ ಕಾರ್ಯಕ್ರಮ ಇರಬೇಕು, ನನ್ನದೊಂದು ಗೋಲ್ಡ್ ಮೆಡಲ್ ಇರಬೇಕು ಅಂತೇಳಿ ಬಹಳ ಗೌರವದಿಂದ ಇಲ್ಲಿ ಅದನ್ನ ಇನ್ಸ್ಟಿಟ್ಯೂಟ್ ಮಾಡಿರ್ತಕ್ಕಂಥದ್ದು. ಹಾಗಾಗಿ ಇದನ್ನ ವಿಶ್ವವಿದ್ಯಾಲಯ ಬಹಳ ಜವಾಬ್ದಾರಿಯಿಂದ ನಡೆಸ್ಕೊಂಡು ಹೋಗ್ಬೇಕು. ಹಾಗಾಗಿ ಇದು ಯಾವುದೇ ಡಿಪಾರ್ಟ್ಮೆಂಟ್ಗೆ ಸಂಬಂಧಪಟ್ಟ ಕಾರ್ಯಕ್ರಮ ಅಲ್ಲ. ಕೋಆರ್ಡಿನೇಟರ್ ಅನ್ನ ಮಾಡಿದ್ದೀರಿ, ಅವ್ರು ನಡೆಸ್ಕೊಂಡು ಹೋಗ್ತಾರೆ ಅಂತಕ್ಕಂಥ ಭರವಸೆ ಹೊಂದಿದೆ. ಹಾಗಾಗಿ ಇದು ಮುಂದೆ ಸೊ ಅದಕ್ಕೆ ವಿಶ್ವವಿದ್ಯಾಲಯ ಒಂದು ಒತ್ತನ್ನು ಕೊಡಬೇಕು ಅನ್ನೋದ್ರ ಜೊತೆಗೆ ಈ ಕಾರ್ಯಕ್ರಮಗಳು ನಡೀಬೇಕು ಅಂತಕ್ಕಂಥದ್ದು. ಈಗ ಎಲ್ಲ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ನಾನು ಒಂದು 10 ನಿಮಿಷ ತಗೊಳ್ತೀನಿ ನಾನು, ಯಾಕಂದ್ರೆ ಮತ್ತೆ ಲೆಕ್ಚರ್ ಆದ್ಮೇಲೆ ಮತ್ತೆ ನೀವು ಯಾರು ಕೂರುವುದಿಲ್ಲ ಇಲ್ಲಿ. Indian education system started from Nalanda Takshashila University. You know that. You open the door and you see the website and see what is the status of Nalanda University education was given. What is the type of quality education was given in Nalanda Takshashila universities, and what kind of students are attracted from those universities. ಯೂನಿವರ್ಸಿಟಿಗಳು ಒಂದು ಅಟ್ರಾಕ್ಟಿವ್ ಕೆಲಸವನ್ನು ಮಾಡತಕ್ಕಂತ ಕೆಲಸಗಳನ್ನ ಮಾಡಬೇಕು ಅಂತ ಒಂದು ಶೈಕ್ಷಣಿಕ ವಾತಾವರಣವನ್ನು ವಿಶ್ವವಿದ್ಯಾಲಯಗಳು ಕಲ್ಪಿಸಬೇಕು. ನಲಂದ ತಕ್ಷಶಿಲಾ ಯಾವ ತರ ಒಂದು ಶಿಕ್ಷಣವನ್ನು ಕೊಡ್ತಾ ಇತ್ತು, ಅವತ್ತು ಎಷ್ಟು ಕಂಟ್ರಿಗಳನ್ನ ನಲಂದ ತಕ್ಷಶಿಲಾ ವಿಶ್ವವಿದ್ಯಾಲಯಗಳು ಅಟ್ರಾಕ್ಟ್ ಮಾಡಿತ್ತು. ಈವನ್ ಫಾರಿನ್ ಕಂಟ್ರಿ ಸ್ಟೂಡೆಂಟ್ಸ್ ಆರ್ ಅಡ್ಮಿಟೆಡ್. The subjects were thought, politics, astrology, astronomy, everything. Today, our Honorable Vice Chancellor belongs to the physics subject, and today the talk of the whole world subject is a teaching a physics and astronomy, physics and astrology. You see the relationship, how physics is going to relate it to the astrology. These multicultural practice started long back in Nalanda Takshashila. How the subjects are going to become an interdisciplinary. What is the importance of interdisciplinary? ನೀವೀಗ ಮುಂದಕ್ಕೆ ಬಂದ್ಬಿಟ್ರಿ. ಈಗ NEP SEP ಬಗ್ಗೆ ಚಿಂತೆ ಮಾಡುವಂತ ಅವಕಾಶ ನಿಮ್ಗಿಲ್ಲ ಇವಾಗ. ಆ SEP NEP ನಲ್ಲಿ ಏನಿತ್ತು? ವಾಟ್ ಈಸ್ ದಿಸ್ ಮಲ್ಟಿಕಲ್ಚರಲ್ ಪ್ರಾಕ್ಟೀಸಸ್? ಇಟ್ಸ್ ನಾಟ್ ನ್ಯೂ. Multicultural practices are not new. It was started in Nalanda Takshashila time, not now. Interrelated subjects, understanding politics, understanding economics, understanding medicine, understanding the interdisciplinary of social science, sciences, languages. Today the skill has come because of that purpose only. Learning. We have started 4 pilot experiments in 4 universities now. The subjects are related. Along with your core subject, we are introducing a skill-oriented subjects for the availability of employment. Immediately students should get the employability. What the government has to take an initiative. We were talking Upper Offerdale and myself in the breakfast. Education has to divert towards the employability. Everybody is studying the purpose of employability. How employability will create? Whether these skills are going to create an employability? In that case, the Karnataka Skill Development Corporation has come forward to calling and tender for the industries who are giving the skill-oriented courses. 11.52 lakh students have taken the UPSC examination. The passing percentage is 0.5%. 25 lakh students have taken the examination of KPSC in 2019. The passing percentage is 0.2%. Why our students are not excelling in competitive examination? Where is the writing skill? Where is the writing skill and understanding skills and disseminating the knowledge on the piece of paper? Why this percentage? That's what government is thinking, that our Indian students are not able to compete with the competitive examinations. Employability skill, cyber security is one of the one. And recently I attended the World Education Forum in London. 140 countries were represented there, and whole discussion in World Education Forum was the artificial intelligence and machine learning. Once the artificial intelligence is entered to the education system, man has no work. Man has no work. I don't know how the universities are going to adopt a system for the work culture with this growing population. Artificial intelligence and machine learning will give a new avenue for the learning in all kinds of disciplines. We are moving towards that side. Your publications. 1.3 million papers were published in India, compared to 1.4 million papers in UK, 4.52 million papers in United States, and 4.6 million papers in China. When you take the H-index of the United States is 47.5. 39.5 of China. UK is higher than the India, and our H-index is 14.07. My dear students, keep it in your mind. Why our H-index is down? Why the United States papers are quality papers, and our research is 75% is the substandard papers were published. That is a quantitative research. And the qualitative research is the 14%. Out of 100%, 75% is the quantitative research, and the 14% is the qualitative research. And 5% is the mixed research, and the rest of the percentage is environmental kind of research. So publications, impact factor, and research, quantity and quality, if we take, these are all the best challenges and the right challenges in front of us to bring the students to publish a quality papers. If you are not publishing a quality paper, if you are not doing some kind of a cut-edge research, then quality papers will not come. If you take the whole University of Mysore paper H-index, I will not spell out, you right to see that. You are the right person to see where you are now. What is your status as far as world is concerned? Each and every university has a rating in publication quality and the quantity. That you see that there is some declination of the university. 100-year-old university, there is a declination of the research. That is the caution for us. You have to take this university and publish a good number of papers. Then we can cherish better to enter. Why the Indian students are not getting the state seat in Oxford and Cambridge? Even today, no one university is listed in first 200 universities in the globe. The reason behind is 75% of a quantitative research. Research has to be uplifted and universities has to take. We are working for the Indian higher education landscape is a mix of progress and challenges. Its scope is vast. 1,200 universities, 42,000 colleges, 11,000 and standalone institutions. In Karnataka, we have 32 universities, 1,32,000 students. All together, medicine and agriculture, we have a 22 lakh students in the state. This is skill-oriented program, and artificial intelligence, your cyber security, your global exposure, your digital learning, all these areas are coming up. Now the state government has taken a lot of initiative in this area to put you people in the employability education. Of course, you people are going out, but whether you are getting that opportunity or not, but government is having a large scale, large long-term education system the employability in Karnataka. So governance and education has to go together. Education, now the higher education information is now right age is the transformative age. This is the transformative age now. From conventional system of education to advanced system of education. Now we made it we called it as a restructured curriculum for the NEP. We are not calling it an state education policy or national education policy. Government is thinking in restructuring of a existing curriculum for the undergraduate and postgraduate courses. And restructuring, and we are keeping the UGC guidelines in the mind. State government has come forward to maintain the standard of UGC guidelines, and we are putting that a restructured education system. So this education age is 4 now. 1 is completed, 2 is completed, 3 is completed. We are in the education 4 in India. You should know what is that 4, what is that 1, what is that 2. You must know, my dear students, no need of teacher here afterwards. That doesn't mean ಇವರೆಲ್ಲ ಪಾಠ ಮಾಡಬಾರ್ದು ಅಂತ ನಾನು ಹೇಳ್ತಾ ಇಲ್ಲ, ನೀವು ಕೇಳಬಾರ್ದು ಅಂತ ನಾನು ಹೇಳ್ತಾ ಇಲ್ಲ. But how long you will depend on the teaching alone? You have to expose yourself, and you have to see that what is going in the globe. That's called as a global exposure. What kind of global exposure you have in your mind? Now the learning is, the sky is the limit. You have a new gadget with you. That's the computer. Your mobile is the best teacher for you today. Provided if you use properly. Provided if you use properly, this computer and mobile is the best teachers for you. Don't you think like that? Another 2 decades, 3 decades. Today, computer science students, every engineering student's top subject is the computer science. I don't know what is the mindset of the students. A mechanical engineering student and a civil engineering student is shifting for MBA program. What is the duty of a civil engineer? What is the duty of mechanical engineer? What is the duty of electrical engineer? So they are going for master business program. Why? Why this? Because everybody wants to become a multi-millionaire to make the marketing and business. A product development, science and technology. We have succeeded, my dear students. Science and technology will take a different total arena. If I go on talking on the development of science and technology, India is enormous. It is unimaginable. Starting from your satellite, vaccines, green revolution. 1947, hardly we don't have any food in this country. When Jawaharlal Nehru took a charge as a prime minister, hardly there was no communication. Food security was not there. Poverty was high. For first 13 years up to 1960, when Swaminathan brought the green revolution, until that time we suffered, our elders have suffered, our parents have suffered really without a food security was not there. M.S. Swaminathan was selected as a police officer, we know that. He refused to become a police officer in UPSC. And he is a genetic, a breeding man, and he said that I want to become a scientist of this country. My country is suffering with the food security. I will sustain that food security, and he is invited in Norman Borlaug, and he sustained the food security in this country. Today we don't have dearth for the food another 30, 40 years. Dr. Vasan Kumar sitting here, he agree with me. Agriculture is planned in such a way. We are producing 50 million ton grains. Today we are producing 400 million tons. We don't have a dearth for the food. ನೀನು ಕೆಲಸ ಸಿಗುತ್ತೋ ಬಿಡುತ್ತೋ ಗೊತ್ತಿಲ್ಲ, ಈ ದೇಶದಲ್ಲಿ ನಿಮ್ಗೆ ಊಟ ಅಂತೂ ಸಿಗುತ್ತೆ. You will get food. But next challenge is the education. How to sustain the education, how to improve the education, how we are bringing the policies, how policies are going to help for the young generation, whether these skill courses are going to help the generation. And today, the talk of the town is artificial intelligence and machine learning. How this artificial intelligence and machine learning is going to help for our skill, help for our technology, how to sustain these technologies. We have developed a vaccine, doctor is sitting here. 1-2 ವರ್ಷ ಮುಂಚೆ ನಾವು ಯಾರು ಒಬ್ರಿಗೊಬ್ರು ಮುಖ ನೋಡಂಗಿರ್ಲಿಲ್ಲ ಅಲ್ವಾ, ಒಬ್ರಿಗೊಬ್ರು ಕಣ್ಣು ನೋಡಂಗಿರ್ಲಿಲ್ಲ, ದೂರ ನಾವೆಲ್ಲ ವಿ ಆರ್ ಲೆಫ್ಟ್ ಔಟ್, ಉಳ್ಕೊಂಡವ್ರು ನಾವು ಸಾಯ್ದೆ ಅಂತ ಪರಿಸ್ಥಿತಿ ಈ ದೇಶದಲ್ಲಿ ನಿರ್ಮಾಣ ಆಗಿತ್ತು. ಅಂತ ಸಂದರ್ಭದಲ್ಲಿ ನಾವೊಂದು ವ್ಯಾಕ್ಸಿನ್ ಅನ್ನ ಮಾಡಿಕೊಂಡು ನಮ್ಮನ್ನು ನಾವು ಸಸ್ಟೈನ್ ಮಾಡ್ಕೊಂಡ್ವಲ್ಲ. ಡೋಂಟ್ ಯು ಥಿಂಕ್ ದಟ್ ವಿ ಆರ್ ಪ್ರೌಡ್ ಇನ್ ಡೆವಲಪಿಂಗ್ ದ ಟೆಕ್ನಾಲಜಿ? Now is the duty of how to sustain the green revolution, how to sustain the vaccine technology, how to sustain the satellite technology, how to sustain the drug discovery and drug development. So these are all the success stories of a science and technology development. So in that, I don't want to take much of your time. Still there is a lecture is there and Vice Chancellor is going to speak on the occasion. My dear students, you have a big responsibility. Don't try to manage the education. Don't try to manage the science. Don't try to manage the research. Don't try to publish a paper who belongs to 75% of a quantitative research. You want to belong to the quantitative research of 75% or the qualitative research of 14%? ಯಾವುದ್ರಲ್ಲಿ ಇರ್ತೀರಿ? 75% ಅಲ್ಲೇ ಉಳ್ಕೋತೀರಾ ಅಥವಾ 14% ಕ್ವಾಲಿಟಿ ಅಂತ ಇದೆಯಲ್ಲ ಅದರೊಳಗೆ ಬರ್ಲಿಕ್ಕೆ ನೋಡ್ತೀರಾ? ಅದರೊಳಗೆ ನೀವು ಬರ್ಲಿಕ್ಕೆ ನೋಡೋದಾದ್ರೆ ಇಟ್ ನೀಡ್ಸ್ ಲಾಟ್ ಆಫ್ ಮೈಂಡ್ ಸೆಟ್ ಅಂಡ್ ಕಮಿಟ್ಮೆಂಟ್ ಟು ಪಬ್ಲಿಷ್ ಎ ಕ್ವಾಲಿಟಿ ಪೇಪರ್ ಇನ್ ದಿ ಯೂನಿವರ್ಸಿಟಿ. Do research wherever facilities are available. Don't demand the vice chancellor, don't demand the registrar, don't demand any professor for the infrastructure. You run wherever the infrastructure is available in the country. If you want to do a research, a Nobel laureates who are in the back, you take it, Einstein has not read anything. Einstein has not read anything. They are fully uneducated fellow. The technology has come. Only thing is we need the skill and mindset and scientific temper. That's what the last word I wanted to tell you is, Jawaharlal Nehru said in the beginning itself of the country after taking the charge as the Prime Minister, only science and technology can remove the poverty and hunger of this country. Only science and technology can remove only science and technology and education can remove the hunger and poverty of this country. This statement was made by Jawaharlal Nehru, none other than the first Prime Minister of this country. So this, keep it in your mind, the technology and the education is very important for your sustainability. And Prof.H. Shekar Shetty, the man like Prof.H. Shekar Shetty or anybody who was sitting as a scientist, they are the inspiring models. They are the inspiring model to take that as the part of this thing. Don't struggle for unnecessary things. Don't fight for unnecessary things. That is a waste of time with you. That's a waste of time. You are wasting your time. ಅದಕ್ಕೆ ಒಂದೇ ಒಂದು ನಿಮಿಷ ಸರ್, ಒಂದೇ ಒಂದು ಕಥೆ ಹೇಳ್ಬಿಡ್ತೇನೆ. ಒಂದು ಡಾಂಕಿ ಅಂಡ್ ದಿಸ್ ವಾಟ್ ಇಸ್ ಹೇಳ್ತಾರಲ್ಲ ಹುಲಿ, ಟೈಗರ್ ಅಂಡ್ ಡಾಂಕಿ ವಾಸ್ ಫೈಟೆಡ್ ಈಚ್ ಅದರ್. Donkey said the grass is blue. Tiger said how the grass is going to become blue? Of course it is green. Grass is green. And donkey started to argue like anything, shouted, no, grass is blue, grass is blue. Then tiger is very sensitive. Then tiger kept quiet. No, it is not correct. Let us go to lion. Both of them, they went to lion for the judgment. Then donkey again shouted in front of lion, you see, my lord, this is telling that the grass is green. I said grass is blue. And again donkey started shouting and lion said, yes, Mr. Tiger, grass is blue. And then donkey said, punish him. Then punishment, 3-day silence. That is the punishment for tiger. It is the 3-day silence for tiger. Both animals, they went. Then tiger went back to lion. My lord, of course grass is green. Why you give such a judgment for that donkey? Donkey was shouting like anything. And you said the grass is blue. Yes, I know grass is green. But arguing with the fools is a waste of time. Arguing with the fool is a waste of time. And you are the tiger. Why you have started arguing with the fool of donkey? So of course you are right, grass is green. So arguing with unnecessary things, you are going to become fool and wasting of time. You see that? Make use of your time and take this as a model and grow yourself and establish yourself. Come up in your country, in your life, and life is beautiful provided if you make it. Thank you very much. Thanks, Amrutesh. I wanted to tell 2 words with Amrutesh. Amrutesh has organized this lecture and very beautifully, and I thank them. He was invited for this occasion. I have no words for that because when Shekar Shetty name comes, so I cannot say no in any occasion. I agree, and he has organized very well. And thanks for inviting Amrutesh. Amrutesh has given the responsibility by the university. Whoever gives the responsibility to the university, your duty to do this job. And university is giving a bread and butter for you. Whatever the university directs, you are bound to do it. You are bound to do it. You are a subordinate to the university. And we are all working for the university. Thank you very much.
Thank you, sir. On behalf of the organizers, I now request our Honorable Vice Chancellor to felicitate Professor S.R. Niranjana, the Chairman of KCGC. Moving forward, the students of Prof.H.Shekar Shetty, deeply inspired by his teaching and guidance, have united to create a lasting tribute. They aim to establish a new endowment at the University of Mysore in his name, ensuring that his legacy continues to inspire future generation of scholars. To officially announce this noble initiative, it is my pleasure to invite Professor K. Amrutesh, the coordinator of University Endowment Lecture Organizing Scheme, to share this momentous news with all of us.
Dr. Nandini, Dr. Janathan, I request to join. Please come. With the permission of Honorable Vice Chancellor and the other guests that are there in the audience, I am extremely happy to announce. Actually, when I wanted to institute, we the students of Professor Shekar Shetty, another endowment lecture in his name, especially in the name of Pulmonary Downy Mildew Research Laboratory. He has expressed, no, my guide name should not be neglected. That is the founder of Department of Applied Botany and Biotechnology, Professor K.M. Safiullah. So that's why we, the students of Professor Shekar Shetty, wanted to have Professor K.M. Safiullah also. Henceforth, we are extremely happy to announce Professor K.M. Safiullah and Professor H. Shekar Shetty Pulmonary Downy Mildew Laboratory Endowment to the University of Mysore by donating 5 lakhs. So this is another occasion for all of us. I request all the Professor Shekar Shetty students to join us, including Dr. H.S. Prakash and Dr. D. Vasant Kumar. Please come and join us in handovering the rupees 5 lakhs to our Honorable Vice Chancellor to mark special lecture every year in the name of Professor K.M. Safiullah, who established international reputed laboratory that is nucleus of the department, that is Pulmonary Downy Mildew ICR program. Even today it is running successfully, and that's why. So we would like to institute the major lion's share given by Dr. G. Chalvaraju, who is here, who is a global head in one of the companies. So other students have also joined in this program. We are donating rupees 5 lakhs in the name of again Professor K.M. Safiullah, the mentor of Professor Shekar Shetty, Professor Shekar Shetty Pulmonary Downy Mildew Endowment. I request our Honorable Vice Chancellor to accept our rupees 5 lakhs in the name of Professor K.M. Safiullah, Professor Shekar Shetty to remark.
1 by 1 ಬನ್ನಿ ಎಲ್ಲರೂ ಬರ್ತೀವಿ ಸರ್ ಹಿಂದೆ ಸ್ವಲ್ಪ ಹಿಂದೆ ಹೋಗಿ ಥ್ಯಾಂಕ್ ಯು
So at this juncture, with the permission of our Honorable Vice Chancellor, we would like to greet our Professor Mentor, Dr. Shekar Shetty on the occasion. Please, sir, I request Vice Chairman of the Karnataka State Higher Education Council, Professor Podile, and our Honorable Vice Chancellor to conduct this event, to felicitate our Professor Shekar Shetty, please.
ಇಲ್ಲ ಒಂಚೂರು ಈ ಕಡೆ ತಗೋ ನಾನು ಆ ಕಡೆ ಇಲ್ಲ ಇಲ್ಲಿ ಬರಲ್ಲ ಹಾಂ ಹಾಕಬೇಕು ಸರ್ ಈ ಕಡೆ ಕ್ಯಾಮ್ರಾ ನೋಡಿ ಸರ್ ನಿರಂಜನ್ ಸರ್
Thank you, dignitaries, for doing the honors. Ladies and gentlemen, it is with great pleasure that I stand before you to introduce the distinguished academician and researcher, Professor Apparao Podile. With over 35 years of exemplary teaching and research experience, Professor Podile's contributions have left an indelible mark on the field of molecular plant microbe interactions. Professor Podile served as the Vice Chancellor of the University of Hyderabad and continues to inspire as a senior professor. Beyond his academic achievements, Professor Podile is a fellow of all major science academies in India, highlighting his stature in the scientific community. He is an elected fellow of Indian National Science Academy, Indian Academy of Sciences Bangalore, National Academy of Agricultural Sciences New Delhi, and National Academy of Sciences Allahabad. His accolades include prestigious fellowships and awards such as J.C. Bose Fellowship and Tata Innovation Fellowship. Professor Podile was a Fulbright Fellow in the Academic Leadership Seminar at Cornell. He has also served as the chairman of the southern region of AICTE. His commitment to advancing scientific knowledge extends to influential roles on national level advisory boards and esteemed scientific councils, where he contributes significantly to shaping scientific policies and education. Currently, Professor Podile continues to lead cutting-edge research endeavors supported by substantial grants, further advancing our understanding of plant microbe interactions. Now, on behalf of the organizers, I humbly call upon Professor Apparao Podile to deliver this year's Professor Shekar Shetty Endowment Lecture. Sir, the stage is yours.
My colleagues on days with me, Professor Niranjana, the Vice Chairman, Karnataka State Council for Higher Education, Professor Loknath, Honorable Vice Chancellor, University of Mysore, Madam Shailaja, Registrar of University of Mysore, and my respected teacher, Professor Shekar Shetty, and a good friend, Professor Amrutesh, several faculty members from the different departments of University of Mysore, and a very good friend of mine, Dr. Vasant, and my dear students. For me, joining you all today gives me immense joy, an immense joy for a variety of reasons. To join you on an occasion like when the university is celebrating H.S.Shekar Shetty H.S.Shekar Shetty Endowment Lecture as a very, very special occasion, I must thank the pioneer those days, DuPont, subsequent DuPont, the sponsors of the event, the sponsors of the endowment, for thinking about instituting this kind of a endowment lecture in the name of a great innovative researcher like Prof.H.Shekar Shetty. I think the Manas Gangurti campus, probably the greenery of this or the water that you drink on this campus has something in it, because the people I have known from this campus, all of them have exceedingly done well in their own disciplines. That shows that there is something very special in this campus, sir, and for which you are the custodian today. Therefore, I really feel honored to be at University of Mysore. This may be maybe 5th time or 6th time I have come, but I always feel happy to be at University of Mysore, and especially on an occasion like this. This university has a wonderful character, in my opinion. A university which has a legacy of over 100 years has nurtured humanities, especially the languages, and the other subjects of social sciences and sciences equally, and has been an excellent model university for several of the states in the country, and therefore this stands as a unique university, and the people who have contributed to this development of this kind of a university must be appreciated for all that what they have done and brought to this level today, where you are remembered or you are recognized as one of the best universities in the country. I congratulate all of you. I congratulate leaders like Professor Safullah, Professor Shekar Shetty, Professor Rangappa, Professor Rangana, several others, several names that I can take off. Many of you have been role models for the higher education in the country, and you have brought this university to this level. So just before I start the lecture, I have to tell a couple of things, and then I will go to the lecture part. Honestly speaking, about University of Mysore, for the first time I have known more was it when I met Dr. Vasant and Dr. Sudhir Shetty and Dr. Sharada. Three of them were co-participants at Banaras Hindu University in a conference organized in Department of Plant Pathology. Vasant was a faculty member by that time. I was just a student. And that was the time I have come to know about University of Mysore and the kind of research that is being done at University of Mysore. And since then, until today, with the generations that we have seen now, Amritesh has come up, the flow. Don't you see that I am speaking very slow, and compared to Amritesh and Niranjana, they speak with fluency. They speak with some kind of authority. But I am speaking very slow compared to them. That is what speaks about the new generations have come and they are doing so well. And that is the university. And therefore, again, let me tell you, I am very happy to be with all of you here. And when maybe a couple of weeks ago, when I was contacted by Professor Niranjana, about these, informing me about this endowment lecture, and the possibility that I can be the speaker for this occasion, that was a very joyous moment for me, because as you heard somebody saying, Professor Balram gave an endowment lecture, H.Shekar Shetty, Professor Ranjanath gave that talk, Professor Anupam Verma gave that talk, and I am along with them giving an endowment lecture in the name of a person for whom I have highest regards. Therefore, I feel so happy to be all of you today and to share with you some information on what I have known and what is known in the literature, and I will add a little bit of what research that we are doing in that. And for an endowment lecture, I always thought it should be gentle, it should be simple, and a physics professor like Professor Loknath also should be able to take message at the end of the lecture. If he is able to spend the full 1 hour with us, he should also be able to take some message from this lecture, was what my intention was. Obviously, it will be as simple for the biology students if biology students are sitting in the audience. That's the kind of thing that I have planned, and I wanted to talk to you on something what we have not known as a student. I'm sure Vasant and Niranjana will agree with me. Immunity word, plant immunity word was not told to us when we were students. Then nobody would agree that there is something, some word immunity can be used in plants. So that was the status from where we have come, and today we know plant immunity is a major area of science, and there are different levels of immunity in plants, and we will only talk about one level of immunity, because if we talk about all immunities, it will take a long time. Even this one immunity, to convey to you as a concept, and it may take a little longer, you may have to bear with me for that extra few minutes for me to convey the concept of immunity, especially the innate immunity part of it. With this, I will move to the slides, and from there I will speak. Here is the man about whom you all have heard through Professor Amrutesh, through Professor Niranjana, and even otherwise you have known. And a nice gesture was there on the part of his students, again, to institute another endowment lecture, combined with Professor Safullah and Professor Shekar Shetty, and to remember their outstanding contributions in the Downey-Meadow research from the Downey-Meadow laboratory. That was a remarkable thing. I congratulate all the students who have contributed to this endowment, and a great man about whom we are talking. A person whose research, I just was looking at one of the things he has, one of the most highly cited papers from Professor Shetty's lab is role of reactive oxygen species, which is part of plant immunity. And seed biopriming is also a part of immunity, induction of immunity. So the two most cited papers from Professor Shetty's lab are directly connected to the plant immunity. Therefore, I thought I have not done anything wrong by choosing a topic like innate immunity to talk to you as the endowment lecturer in the name of Professor Shetty. You can see outstanding citation record for that speaks. Professor Niranjana was talking about it. You want to be in the 14%, you want to be in the 75%. He is in this category where you can see the world over, people have recognized the kind of research, the qualitative research that has been done by Professor Shetty and his group, and they have highly respected his work and cited at least 10,000 times. So I am honored to be here to speak about, in an endowment lecture that was instituted in his name and by Pioneer. Thank you so much for bringing me here. Well, as I said, some of the things I will go down because my physics professor should also understand tomorrow. He should also tell people that plants have immunity. Okay, so you have plants growing in different places. They grow in forests, they grow in interior, they grow in deserts, they grow everywhere. But wherever they are growing, Unlike as the tiger believed, they look green. As the tiger believed, they look green. Okay, they look so green, so nice, and it is all we are enjoying. And in the morning when I went for a walk in the campus, I really enjoyed the beautiful campus. The number of trees, the kind of trees, that the lush green campus. But they also suffer from diseases. They also suffer from diseases. When they are affected by diseases, some of these plant parts can become like this. The fruits can get damaged like this. The leaf can get damaged like this. And the entire plant can get damaged like this. The blight disease can be something like this, as serious as this. So you have beautiful plants, and also there are diseases which can affect the plants. But then, this is what is the beauty. What we see in the plants, all of them, what we see in this screen, are all beautiful green plants. How is that? When there are pathogens which are causing diseases, Vasant has written a beautiful book about the pathogens. And I really like that book, and how to understand these diseases and how to manage those diseases. So many important things that he has brought into that book. That's another major contribution. That's where I said there is something in this Manas Gangotri campus. So all of you should get inspired for studying in the campus itself. Forget about looking at the teachers. Studying in this campus itself is a big thing for all of you. How these are all remaining green when there are thousands of fungal pathogens, hundreds of bacterial pathogens, and hundreds of viruses. There are so many of them, but still they are looking green, they are surviving. That means they have something in them to prevent the entry, to prevent disease causation by these pathogens. For a minute you will imagine that they are not having any mechanism of their own to prevent the entry of pathogens. The situation will be like this. What is it? No single plant there. There is no single plant. If there was no immunity in the plants, if there was no mechanism by which the plants can survive against the onslaught of the pathogens, infectious agents, particularly the fungi, bacteria, viruses, it would have been like this, It would have been like this. But then, situation is not like that. If it is like this, what would have happened? Niranjana was talking about, we have surplus food, we are able to produce green revolution, that revolution, that revolution. Where is the green revolution if there are no plants? There would not have been any green revolution, there would not have been food for us, and maybe all of us would not have assembled here, we would not have been there to assemble, because we are dependent on plants for that. Therefore, my dear friends, remember that the plants have a mechanism to survive against the onslaught of the pathogens, even if whether you are able to give some fungicide or not, bactericide or antibiotic or not, they still have some mechanism of their own. This is a very, again, Niranjana was speaking, repeatedly he took reference to the artificial intelligence, computer science, the way the people are getting attracted towards that. That's a different story. I'm not getting into that. What I want you to understand is that, I think this is closing it, there is a term called pattern. Pattern recognition. In every aspect that we use today, this pattern recognition has become important. You take your mobile and your face, it recognizes. You put your fingerprints, that is recognized. These are all pattern recognitions. These are all pattern recognitions that are happening, that are extensively used as a technology today, and the living organisms have been using this pattern recognition for millions of years. This pattern recognition is used by living organisms, only the computer science fellows are using today. Living organisms have been using this pattern recognition for ages. I just give a small example here. If we take a banana, if we make the room dark, completely dark, and if we distribute banana fruits to all of you, everything is dark. You will say, you have to identify what is the object that is given to you. 99.9% of you will identify it as banana. just by sensing it from this end to that end. Because of the texture, you may say, because of the shape, you may say, different things. These are all the patterns that are got imprinted in our mind, therefore we never forget that. And that kind of a pattern recognition has been a unique phenomenon in biological systems, not only in plants, it is there in animals as well. So the innate immune system, I'll try to tell, because there is always a confusion, people always thought, teachers like Prof. Shetty will agree with me, plant bacteriologists have always been kept aside by medical microbiologists. There may be medical microbiology students here. Please listen carefully. Medical microbiologists always thought plant microbiology is different and human microbiology and medical microbiology is different. They never thought bacteria are bacteria, Pseudomonas is Pseudomonas. They never thought. The problem comes there, and they thought whatever is seen in animals will not be there in plants. Therefore, these two are different things. But today we know both of them have almost the similar lines of evolution in terms of recognizing the pathogens and in terms of mounting their defense responses against the pathogens. It's a first line of defense against the germs entering the body. It is applied to the plants as well. Responds in the same way to all germs and foreign substances. Because of that, it becomes non-specific. Because of that, it is non-specific. And limited power to stop the germs from spreading. It cannot definitely stop, but it can halt them for a while. It can halt them for a while, and that's what is the limited power. But remember, it is always a rapid response. Very quick response that the plants, that the organisms will do, higher organisms against these microorganisms. The adaptive immune system, I will not go to that because that is the second line of defense, which these are slightly different kind of things. This system takes over if the innate immune system is not able to destroy the germs. Specifically targets the type of germ. Is it a fungus? Which fungus it is? Is it a bacterium? Which bacterium it is? Is it a virus? Which virus it is? And therefore it first needs to identify the germ. And therefore it is slower to respond compared to this than the innate immune system, but when it responds, it's more accurate. Because it is completely governed by a gene for gene interaction. A very specific gene for gene interaction. Don't worry about the wordings inside, don't worry too much on that. What I want you to understand is a comparison that is made between plants and animals. The pink is animals and the green is plants. The pathways should look similar. Whatever recognition that happens on the surface looks similar. The kind of responses the molecules that are involved look similar. And the kind of pathways that are activated are similar. The outcomes are similar. Between the plants and animals, the kind of recognition process, the kind of molecules involved, the kind of signaling pathways activated, the kind of outcome that you see when a higher organism is interacting with a microorganism which is able to cause a disease. We'll see more details as we move forward. So I wanted to tell you that this immune system of plants shares similarities with the innate immune system in animals and lack an adaptive immune system. Remember that. As of now, we don't have strong evidences to show that the plants have an adaptive immune system. Plant immunity can be considered under two different categories. What is cell surface? Plant cell, and on the surface. Recognition happens on the surface itself. And another one happens inside the cell. So innate immunity has two components. The cell surface interaction with the pathogen molecules and the intracellular, inside the cell, the molecules interacting from inside the cell. So the cell surface immune receptors detect common signatures of the pathogens outside the host cell via extracellular domains. Don't worry too much. As we move forward, you will understand each one of them. And innate cellular responses to resist infection via their intracellular kinase domains. So that means if there is something which is coming from if this is the cell which is coming from on the top, this is something to recognize on the surface, something to activate inside the cell. Such kind of patterns that are recognized by micropathogen by the plants emanating from microorganisms, in a normal sense we call them as microbe-associated molecular patterns. Microbe-associated molecular patterns. And to be more specific, when they are originating from pathogens, we use the word pathogen-associated molecular patterns, or PAMs. So the MAMs are, MAMs, also there is another term, I will not confuse you with the DAMs, damage-associated molecular patterns, can be received by the receptors on the surface. These are the receptors that we are talking, the pink ones and the green ones, and one is shown as the big one and another one is the small one. There are some which work independently, and some of them recruit a co-receptor. They depend on a co-receptor and they work together. That's where you find something like two components interacting. Both of them can be identical, both of them can be dissimilar when they are functioning together. This is what is the interaction of these patterns with the receptors and the intracellular kinases that I am talking, the oval-shaped structures inside the cell are responsible for cell surface immunity. But some of you who have studied plant microbe interactions, you would have known pathogens are also clever. When they realize that the patterns are getting recognized, they have evolved alternate mechanisms by which they can cause a disease. They can take over the plant. That is, by secreting molecules which can directly enter into the plant cell, by injecting them like an injection. Injectosome we call it. And when they inject that, these effector molecules, we use the word effector molecules for that. And these effector molecules, they not only suppress the cell surface immunity, and because of that suppression, the plant is not able to express the cell surface immunity. But the plant will not keep quiet. It has evolved an alternate structure, the NLRs. I will elaborate on that after a few minutes, which interact with these effectors and express an intracellular immunity. If the pathogen is clever to subvert the cell surface immunity, the plants are also clever to evolve an alternate mechanism of an intracellular immunity. So a subset of cell surface immune receptors sense damaged self as surrogate for the presence of the pathogens, that the damage-associated patterns. Intracellular immune receptors detect translocated proteins, the ones which are injected into the plants, as effectors which are delivered inside the cell by adapted pathogens, and activation of intracellular immunity is a more robust response. I told you, the first one is a little rapid response, but it is not very specific. But this one is a more robust and more specific response. I have already introduced to you these terms, the pattern, the kind of immunity that is triggered by the microbe-associated molecular patterns, we call it as pattern-triggered immunity, and the counterpart to interact with the microbe-associated molecular pattern is the pattern recognition receptor. So there is a PRR, we will see in the next slide. There will be PRRs. These are all MAMs. What are all the molecules which are getting recognized? Flagella. Many of you know bacteria have flagella. Flagella is made up of a protein called flagellin. Translocation elongation factor 2, lipopolysaccharide, peptidoglycan, chitin, chitosan, lipopeptides, and beta-glucans. Excepting one or two, almost all of them are interestingly carbohydrates. Excepting one or two, interestingly, almost all of them are carbohydrates. Carbohydrates coming from pathogens and a few proteinaceous molecules serve as the kind of MAMs or PAMs for the PRRs to recognize and activate the innate immunity. Primarily, we are talking about cell surface immunity. last sentence you must read. Most of these MAMs are also known to be recognized by the innate immune system in mammals. It's not that plants have differently evolved. Animals, humans, and plants also have evolved on similar lines when they are interacting with the pathogens. This is the kind of receptors that we are talking of. You have the extracellular domain, ectodomain, this is what we are calling, this is the membrane outside the cell, an ectodomain, and a transmembrane domain, and a kinase domain in intracellular domain. When this kinase is not there, it is not going to be easy for this molecule to convey the message of interaction. There are certain receptors which are only proteins, receptor-like proteins we call them. They don't have a kinase domain. They only have an extracellular domain. In such case, they need to partner with a receptor which has a kinase domain, and combinedly they interact with the pattern and then convey to inside of the cell of the plant. Different kinds of pattern recognition receptors that are identified for these loose-enriched repeat. The proteins, a protein-protein interaction in biological systems are primarily mediated by loose-enriched repeats. And a proteinaceous molecule coming from pathogen which has an LRR domain, these are the molecules which are known to have LRR domain. Others are all proteinaceous, carbohydrates. The carbohydrates are recognized by three different types of receptors. The lysine module containing receptors, which we are going to talk more, and EGF and lectin. And these are the ones which we are going to talk more today, carbohydrate receptors, because I said chito-oligosaccharides. Because there's no point in talking so many things. Take one as an example and talk about it. The two major components are receptor-like kinase and receptor-like proteins. There can be a receptor-like kinase or a receptor-like protein which can interact with the PAM. RLKs and RLPs also have other roles in plants. Remember that plants are not only fighting with pathogens. Plants have to do so many things by interacting with the environment. They have to sense if there is an increase in temperature. They have to sense if there is an increase in salt concentration. They have to sense if something else, freezing temperatures are there. So many things it has to do. For all that, they have different kinds of receptors to sense, and these are only a small part of all those kind of receptors that are there in the plants. And they are also important in growth and development, reproduction, and abiotic stress tolerance, symbiosis also. At some stage, I will tell you how a rhizobium, which also has a flagellin, and a pseudomonas, which also has a flagellin, are differentiated by the plant. Rhizobium is a symbiont, also has a flagellin. Pseudomonas also is a bacterium which has a flagellin, but plant allows the rhizobium to come in and Pseudomonas to get out. So many of them are identified. I will not go into these details. Different kinds of pattern recognition receptors identified. You can see a series of them and subfamilies, ligands with which they interact, and the plants in which they are reported. You will see most of them are in Arabidopsis or rice. Arabidopsis, model system, rice as an important crop and also as a model system. It's continuous. That means today we have so many pattern recognition receptors interacting with the microbe-associated molecular patterns recognized in the plants, which are functioning in plant immunity. So many of them. Okay? Now, cell surface immunity is frequently, but not always. Ligand sensing cell surface receptors require co-receptors, I mentioned to you, to transduce the perception of non-self into a response. Otherwise, how the cell will respond? The role of membrane lipids in sensing also has been emerged. I will not go into the details of that. That is a new area that is emerging in the last 2-3 years, or maybe 5 years. More is to be understood. Essential components of the pathogens range from cell wall constituents or subunits of bacterial flagellin or molecules secreted into the apoplast, very important compartment in the plant. That is the place where most of the pathogens interact with the plant. and that's the place where the plants fight with the pathogens to secrete proteins intended for the host cytosol. This is where I was telling Professor Shetty's first highly cited paper, reactive oxygen species. He talked about it. So these specific ligands are perceived by the cell surface receptors at nanomolar concentrations, not even millimolar, micromolars, at nanomolar concentrations, and initiate signaling cascades. That's what are the pathways that I said inside, including the production of reactive oxygen species, cytosolic calcium bursts, activation of MAP kinases, and changes in the expression of various defense-related genes. If I look at the publications coming from Department of Applied Botany, probably every fourth paper is talking about the defense-related proteins expression, the kind of reactive oxygen species, several of the things, the seeds sown by the Prof.H.Shekar Shetty, which are sprouted seedlings, and they are growing in the form of his students and doing such kind of good work on plant immunity. The cell surface immune responses are less volatile when compared with intracellular immunity, do not result in host cell death. The cell surface immunity does not lead to suicidal cell death of the plant, whereas intracellular immunity leads to suicidal death. The plant cell, single cell, or at the most 2 or 3 cells commit suicide to save the remaining part of the plant, what we call as hypersensitive response. However, they constitute an effective host strategy against infection, leading to broad spectrum resistance. That's where most of the time when we are applying some of these agents, they confer some level of resistance to to different types of pathogens. It may not be consistent with all the time, with all of them all the time. You will find that invariably to some level. I'm sorry for this. This is not so clear here. What you can see is these are the molecules, patterns recognized by the plants: chitin, chitosan, peptidoglycan, glucan, mannan, lipopolysaccharide. All of them are carbohydrates. And all of them are also recognized in mammals. That's what I wanted to show through this slide, and not more than that. Now, I shift to the chitin and chitooligosaccharides, and after that, the last part will be a little bit of what we have contributed in this area of research. Chitin is a representative of the fungal MAP. We have been talking about it, and triggers different signaling in wide range of plants. The chitin receptors identified are chitin elicitor binding protein, CEBIP, chitin elicitor receptor kinase, SERK1. These are the two chitin receptors that are identified, CEBIP and SERK1, on the plasma membrane, which have a lysine. That is, the pattern recognition receptor should have lysine modules for it to recognize a carbohydrate ligand. These molecules play an important role for defense responses in rice and Arabidopsis, strictly recognizing the size and acetylated form of chitin oligosaccharides. A little bit of complexity. If there are students from chemistry or organic chemistry sitting in this room, you will understand. We will explain carbohydrates in terms of degree of polymerization. That is, the number of monomeric units that contribute to the polymer. If there are 10 units, we call it as a decamer. If there are 5 units, we call it as a pentamer, and we may have 100 units, 200 units, 500 units for a polymer. Oligomers, monomers, oligomers, polymers. And that means the size plays an important role in this process of recognition. And when it comes to chitin, what is also playing an important role is chitin is a polymer of N-acetyl glucosamine. If it is partly deacetylated, we call it as chitosan. If it is partly deacetylated, we call it as chitosan. So, the degree of acetylation on an oligomer is also important in recognition. Here comes the chemistry. Here comes the chemistry. Not that every molecule can do all the jobs that what we are looking for. These molecules, the lysine receptors also play major roles in signaling in root nodule arbuscular mycorrhizal symbiosis. I told you, we will come back to this point at some place. Lysine receptors are recognizing carbohydrate modules coming from microorganisms. So, if a carbohydrate module is coming from a rhizobium or arbuscular mycorrhizal fungus, the plant has to respond differently. It cannot throw it away, then there will be no symbiosis. I will not bother you again. You can use this, the reference is given here, you can download that information and see. The multiple ways in this chitin and chitin oligomers play an important role in host pathogen interactions in plant pathogen systems. Chitin and its oligomers, especially in the apoplast. Just for a quick information, the plants secrete chitinases and glucanases into the intercellular spaces, which are commonly called as pathogenesis-related proteins. Some of these pathogenesis-related proteins secreted into the apoplastic region. When they are interacting with the fungal pathogen in the apoplastic region, oligomers get released. And those oligomers get recognized by the PRRs and defense is mounted by the plant. So the plant is first sending enzymes into the zone where the pathogens are likely to come and recognize them, break their molecules and recognize those molecules and activate and mount defense against the pathogen. Pathogens are also clever. When the plant is doing this kind of a job, they try to produce some kind of molecules which will block this pattern recognition receptor so that it is not recognized. Beautiful things happen. That is all narrated here in this slide. That's all we are talking here. I'll skip that. It's a representative, we talked about this. Plants can perceive, sorry, this got repeated. Okay. An oligomer, size-dependent chitin sensing mechanism is conserved across plants and mammals. Receptor like cytoplasmic kinase, 7 members are essential players in signaling. Arabidopsis and rice SERP1 proteins play multifaceted functions beyond chitin signaling. Not only that, they have something else also to do and may mediate the crosstalk between chitin signaling and symbiotic or other biotic or abiotic stress signaling. Many of us get confused with so many things that are happening in the plant when you treat with this, when you treat with that, when you treat with that. There is a crosstalk here, my dear friends. There is a crosstalk because the receptors are similar and they are very close in terms of recognizing those molecules. In rice, the kind of molecules that are identified are defined in terms of oryza sativa, OS, CBIP, and SERP1. And you can see here what happens. The chitin oligomer is recognized, this is in the case of Arabidopsis thaliana SERP1. Here it has SERP1 interacting, and here the chitin molecules oryza sativa, CEBIP, chitin elicitor binding protein interacting, and they are transducing the message through this kinase domain. SERP1 in Arabidopsis, SERP1 was identified as an essential lysine module containing receptor-like kinase, RLK. I am using now short forms. Lysine RLK to activate chitin signaling through 3 orthologs. Let's not bother too much. Chitosan is also known to induce defense responses in Arabidopsis through SERP1 independent pathway. SERP1 independent pathway, the PRRs for chitosan signaling are still not known. We are working with chitosan, we still have not looked at, able to identify the chitosan receptor. Chitin receptor is identified. This is the activation pathway. What you can see here is activation model of oryza sativa SERP1, CEBIP SERP1 complex by chitin haptamer. The chitin haptamer interaction will lead to, when it is interacting, will lead to the kind of activation of the MAP kinase pathway and finally to a defense response like this. This is more elaborate, defining individual components of that. Again, I said you can look at this reference for more details. So, finally, what we are trying to understand, this chitin oligomer and these receptor molecules, lysine, these are the pentagons that are shown here are the lysine module containing extracellular domains, and these are interacting with this chitin oligomer and transducing through a phosphorylation signal, and which goes through the MAP kinase pathway, activating the plant immunity, which we are calling as cell surface immunity. I will now switch to the last part, that is what we have done. The cell wall, for some of you who have been doing microbiology, antibiotics, the most common we can take as penicillin. Penicillin blocks the biosynthesis of cell wall. That's where it has become an important molecule for therapy to treat the bacterial infections. That means cell wall is a very important structure for the pathogen. Whether it is in bacteria causing diseases in humans or animals or plants, or whether it is a fungus which is causing diseases in plants and humans. So cell wall is a very important structure that they have evolved, both fungi and bacteria. And in absence of the cell wall, they will not be able to do their activities, excepting some of those wall-less prokaryotes, which are mycoplasmas and some other, they have different mechanisms. Let's not get into that. So cell wall has become a target because it is an important site for the, important structure for the pathogens. Cell wall, hydrolyzed products as patterns. Cell wall hydrolyzed patterns, cell wall hydrolyzed molecules as patterns. If you recall, I was talking about chitin, I was talking about gluten, I was talking about peptidoglycan. All three of them are components of the cell wall of bacteria and fungi. What we have been doing is working with these enzymes, molecules that degrade chitin or chitosan and generate these chitin oligosaccharides and trying to make use of them for agriculture purposes. So it's a very reasonably economical source of, it's not expensive, let me put it that way. It is a source because it is taken from the shrimp shell waste. You can prepare a pure chitin chitosan from shrimp shell waste, like this. And what we have done is we have created a library of chitinases and chitosanases, which can break the cell wall and help us to generate these oligomers. In this, there is a small major component that we have added. That is, one of my students has serendipitously discovered, I would say, that a chitinase which is supposed to hydrolyze chitin was not only doing hydrolysis, but was also stitching together these molecules in a process what he defined as transglycosylation. So, an enzyme, normally we would expect this molecule, if it takes a pentamer, it should release dimer and monomer, or tetramer, or trimer. A trimer, monomer, dimer. But it is not only, it is synthesizing longer molecules than the substrate. That means it is putting them together. That's what is called transglycosylation. So they are being added and they become, the smaller ones are becoming longer. And if you recall, I was talking about the size dependent and acetylation pattern dependent recognition of these oligomers by the plants in activation of immunity. And we come to that, the molecules which have a range of 6 to 8, the oligomers of the size 6 to 8 are more active in inducing plant immunity compared to any other molecules. Okay? A lot of chemistry, a lot of structural biology has been done in terms of their interaction with the receptor and how they are doing all that. That is all there in the literature, you can find it out. And we wanted to, and if you use a chitinase enzyme and then break the chitin, what we get is a dimer and monomer most of the time, which is useless when I want to use it for induction of immunity. And if I go to an organic chemist, a synthetic organic chemist, request him or her to synthesize a molecule of hexamer size or heptamer size or an octamer size, they'll say, sir, it will take a very long time. You have to do coupling, decoupling, so many reactions have to be done. And when we bought these chemicals, remember, 10 milligrams of that we bought for 25,000 rupees. Each of the reference molecules like kytohexose, kytopentose, and kytooctose. It's quite expensive. But we have a mechanism by which the enzyme can do this process, which can synthesize, taking the smaller ones and synthesize the molecules of our requirement. These are all the initial things that we have started way back. Cell wall as a target. Cell wall as a target. Vasant has been listening to some of these things here and there. Many times we met, although he moved to private industry and later he set up his own company, set up his own foundation, so many things he is doing. He still follows what we are doing. So some of these things we have been doing until 2005. Okay? We were not characterizing the enzymes at that time. And I told you, chitosan is soluble, whereas chitin is insoluble. For somebody who is more interested, I will explain in a discussion part or outside the room. Just one slide that I want you to understand. I said that the degree of polymerization is one thing which is important. That's the first dimension for a polymer. First dimension. Second dimension is degree of acetylation. Second dimension is degree of acetylation. That means how many residues are acetylated. Here, all of them are acetylated. Here, only 3 of them are acetylated, then this one is deacetylated. The half of the square that you see in white is deacetylated. Here, 2 of them are deacetylated, 3 of them deacetylated, 4 of them deacetylated. So chemistry of the polymer not only depends on the first dimension, the degree of polymerization, second dimension, degree of acetylation, there is a third dimension, what we can call it as pattern of acetylation. Because a molecule which has 3 deacetylated residues need not be exactly in this way all the time. It can be this way, it can be this way, it can be this way. So there can be a third dimension in terms of its polymeric structure when it is a deacetylated molecule. We have been working on some of these aspects where some of them, depending on the degree of polymerization, we have shown that some of them which have a lower degree of polymerization are antimicrobial. Some of them which are higher degree of polymerization and the transition range between electrolytic range to hydrophobic range are having plant strengthening activity. And these which are the larger and higher degree, they are not soluble and not useful. I told you, one of my students, he just joined Geetham University in Bangalore after 10 years of postdoctoral tenure in US as an associate professor in structural biology. He has joined. This boy has worked extensively on this transglycosylation phenomenon of this chitinase, which is produced by a bacterium called Serratia proteomacculans. And here you can see when he has used the substrate DP6, he not only got the products DP5, 4, 3 and 2, he also got 7. It becomes more obvious when he used the substrate DP5, he got 4, 3 and 2, also 6, 7 and 8. More distinct you can see when he has used a trimer. Trimer is converted to dimer and a monomer, and also a tetramer, pentamer and a hexamer, all of them have formed. That was, when he showed first time I said, go and repeat. No, don't tell me, go and repeat. 3 times he has done. And every time he comes with the similar kind of TLC pictures. Then we looked more into details of that, when we were sure that yes, it is happening. We have done, and remember, all this has been done with pure recombinant chitinase that has been cloned and expressed in heterologous host. It's not that any impurities can be there in the enzyme. And that's where we started off in 2012 publishing this in Journal of Bacteriology. Now, subsequent to that rather, these many different chitinases
More than 3 dozens of chitinases we have cloned from bacterial sources, expressed in a heterologous system, and completely characterized these enzymes. We know which enzyme can break the chitin molecule of which size to what sizes. Everything we have information in a precise manner. About 5 to 6 students have obtained PhD in that particular area. And in addition to that, we also have another library of 40 different mutants of these chitinases. I am yet to see another lab in the world which has a rich library of bacterial chitinases and its mutants, as many as 60 and plus, that's what we have in our lab. All of them are thoroughly characterized. These are all the things, and thanks to all these students who have contributed to this work. Some of them are only postdoctoral fellows, and all of them are PhD students. This was Purushottam. Neeraj started long ago. Suma is in US, and Sharma continues to do work, and he is working in field. And Subnarayan is working as an assistant professor in a central university. Madhu Prakash is an assistant professor in my own department, and she is working in a startup. She has a startup working, and this boy is working for ICR as a scientist, and he is in US. Both of them are in US as a postdoctorals. All of them have contributed to this work. These are the mutants that I am talking of. And all of them also have contributed to the development of these mutants. Just a sample of that, Madhu has done extensive work on this mutational analysis. Entire thesis was on one particular enzyme and all mutations, understanding the structure function of that particular protein. That was Madhu's work. Madhu has targeted, when we know that the enzyme is doing transglycosylation, whether we can improve the transglycosylation process, reduce the hydrolysis. We have not been 100% successful, let me tell you. We were partially successful to reduce hydrolysis and to enhance the transglycosylation to some extent, which is not an easy task, but he has achieved. The mutants that he has looked at, different mutations, these mutations at this position 226, 228, 159, all of them converting to these amino acids. You can see most of the cases he was successful in getting enhanced transglycosylation and decrease of hydrolysis. This is more obvious picture. What I would like you to see in this is after 30 minutes, the transglycosylation products were this many in the when we have used the wild type enzyme. When we have used a mutant, improved mutant of enzyme, even after 6 hours, we have transglycosylation activity, and the activity was much more, and the products are DP5 and DP6 we were able to get happily. He has done a few other things. For those who are interested, the protein, he has mapped the entry of the substrate and exit of the product, mapped the protein after carrying out a wonderful study with a chemistry group and with my own teacher. I studied at Sardar Patel University for my MSc, and those days, can you imagine in a botany class, there can be a lecture, again Niranjana was talking about interdisciplinarity. In 1981, when I was a master's student, we had biophysics and biomathematics in our botany class. Professor T.P. Singh, the world famous biophysicist, who is sitting at, like a distinguished professor sitting in applied botany, distinguished professor sitting in applied botany, as a distinguished professor at All India Institute of Medical Sciences, with all his expertise in structural biology. I called him on telephone one day about this excitement that we have with this protein. He could immediately recall who I am. I was his student several decades ago, but he recognized, and he said, no problem, you send your student to my lab. He was telling, wherever there are facilities, go and do. I sent my student. In 2 months' time, he has returned with the structure of that particular protein. And we have done so many things after understanding the structure of that protein. One of it is understanding the entry and exit sites. So the hypothesis that was generated by Madhu was, if we introduce a bulky amino acid residue at the exit site, it may allow the substrate interaction of the enzyme substrate complex for a longer duration, which may lead to enhanced transglycosylation. So to test that hypothesis, he has done two things. One, he has introduced a bulky residue at the exit site so that the time it gets delayed. And also he has introduced a bulky residue at the entry site, wherein there will be less of a transglycosylation or almost activity is lost. That's the result of it you can see here. When he has introduced the bulky residue at the entry site, almost the activity is lost. When he has introduced at the exit site, compared to the wild type, you find a significant improvement in the transglycosylation activity. Well, we have prepared the phyto-oligosaccharides using a semi-preparative HPLC after using a mutant, the mutant Y2428A, and we have generated these products like this, and we have tested these products for different things. On rice as a foliar spray and as a root dip. And the kind of things that we were looking at are accumulation of phytoalexins, oxidative bursts, synthesis of protease inhibitors, lignification, jasminic acid production, and activation of PR protein, most of the defense responses. And foliar treatment induced elicitation response early and sustained longer. That was what we found. This is the method that we have followed. So priming, another paper that Professor Shetty was talking, the second highest highly cited paper from Professor Shetty's lab was on priming. Priming in plants is a different strategy against the pathogens. This is very well known now. And the induction leads to a kind of enhanced defense response upon pathogen attack, what we call it the primed state. Now today, those days when Vasant was working for the seed companies, they were priming, they were not priming, they were coating with the fungicides that has become a standard practice. Now coating with the priming agents also have become a kind of a practice these days. So we have done something else, the last leg of the stalk. I told you that the third dimension in the oligomer. There's the third dimension in the oligomer, that is the pattern of acetylation. So we were the first people in collaboration with German group to identify that the pattern of acetylation on the chitosan oligomer is important for its biological activity. And that was published in a journal which I would never have thought of submitting a paper, what is called JACS. If chemistry teachers are sitting here, they will understand what is this JACS. Journal of American Chemical Society journal, which is a very prestigious journal, which is as close as Proceedings of National Academy of Sciences. Publishing it in that is as good as publishing in PNAS. So that was the paper that we published. The first part of those work we have published in Bioresource Technology. Again, Madhu was responsible for that, for developing this kind of a cell culture based assay with these oligosaccharide mixtures, and then looking at the reactive oxygen species of burst. Look at this, the third dimension that I'm trying to show you. The acetylated residue is the first residue here, second residue here, third residue here, fourth residue here. Not an easy task for us to generate this kind of special oligomers. We depended on several of the microbial chitin deacetylases which are specific in terms of their substrate and in terms of their interaction, the subsites, interaction subsites specific. And then we generated these molecules, and because for us to test the hypothesis, this was required. And we have generated that, and we see now here, what you see here is the molecule which has the acetylation as the non-reducing end, and this one has the highest activity compared to the one which has the one at the other end. That means the pattern of acetylation has an important role in inducing biological activity. the same thing that we have shown. The monoacetylated chitosan tetramer having N-acetyl-glucosamine at the non-reducing end showed the highest priming activity, and the one which has at the reducing end showed the lowest priming activity. The major conclusion from this paper that was in JACS. Well, I will not take all the publications. So many of them were published during 16 to 19. That was the time where a couple of times Niranjana visited our department also, where as a PhD examiner once and as a passing by as a Gulbarga University Vice Chancellor at Hyderabad, he has visited, and I was Vice Chancellor, and this was the time we were publishing these papers. More papers during 2022 from that. This was all overlapping with my Vice Chancellor tenure. 2022, more papers. So I was telling you, let me hold it on for a while. The final conclusion that we are drawing from this cell surface immunity that we have talked in terms of innate immunity in plants, the lysine module containing receptors which are present on the surface interact with carbohydrate ligands and then transduce a message through a kinase domain and activate responses inside the plant. Okay? If you look at this side more carefully, the first two I am talking about as the nod factors and the MIC factors. Nodulation factor, mycorrhizal factors. Nod factor is a lipid chitin oligosaccharide, for those of you who have studied about it. Otherwise, you can look at it. Lipid chitin oligosaccharide is the nod factor. Similar lipid chitin oligosaccharide is the MIC factor. And all through I have been talking for the last 20 minutes, chitin oligosaccharide, which is activating immunity. One side I was talking about activation of immunity here, when I used the chitin oligomer, of course we have not used a peptidoglycan. Here, the microbe-triggered immunity is activated and the pathogen is suppressed. Whereas in these two cases, IA, welcoming the microorganism inside and allowing the symbiosis to happen. The difference between the two is, here it is a simple chitin oligosaccharide, here it is lipid chitin oligosaccharide. Difference is that the chitin oligosaccharide has a lipid substitution. So, if the external molecule coming from microorganism has the lipid content more, or lipid content, it activates a symbiotic pathway. The receptor with which it is interacting is different and activates a symbiotic pathway. If it doesn't have a lipid component and it has only chitin oligosaccharide, it activates PTI. That is the immunity, innate immunity that we are talking, pattern-triggered immunity, the pathogen-triggered immunity, whichever you want to call it. That's where I said the plants know how to differentiate a symbiotic organism versus a pathogenic organism. The pathogens do not produce these LCOs, symbiotic organisms do not produce only chitin oligosaccharides. I'm not talking in air. Vasant will catch me after I go out. What are the products available? Where is it available? This kind of questions he will ask me. These are all the products that are available with the chitosan. So many products in the Indian market. I'm sure he knows more of them, many more of them. And you will have in the global market as well. So many of them are available. But my dear friends, if we know more of the chemistry and if we can prepare molecules or the products with more precise chemistry, the effect can be much more. What we are achieving with kilograms and milligrams or grams of it, we can achieve with micrograms of it. That is what is possible because they are working through activation of innate immunity in plants. And that is chitin oligosaccharides activating innate immunity in plants is going to be a kind of a strategy, and already a strategy, and it is going to be an important strategy in protection of plants against the pathogens. This is something I wanted to convey as a tribute, as a respect to a teacher who has been engaged in priming, engaged in defense responses, and a person who is known throughout the world for his outstanding contributions in plant microbe interactions in general and plant pathogen interactions, more specifically in downy mildew, pearl millet interactions. Thank you so much. Thank you, sir, for doing this. I should thank all these people, all my students who have done outstanding work, all of them, and then my collaborators, German collaborator, my chemistry collaborator, my teacher, Professor T.P. Singh at All India Institute of Medical Science, another chemistry collaborator, and all these funding agencies which have helped. Last but not the least, I would like to thank the organizers, the committee that has been constituted or that is being put in place by the Vice Chancellor for organizing this H.Shekar Shetty Endowment lecture. And for it is their kind gesture that they have thought of me and invited me here to Bangalore and Mysore to enjoy this beautiful weather, beautiful campus, and sharing my thoughts with beautiful audience. Thank you so much. Thank you.
ಸನ್ಮಾನ್ಯ ಕುಲಪತಿಗಳು, ಕರ್ನಾಟಕ ಜೀವನ ಶಿಕ್ಷಣ ಪರಿಷತ್ನ ಸನ್ಮಾನ್ಯ ಉಪಾಧ್ಯಕ್ಷರು ವೇದಿಕೆ ಮೇಲೆ ಆಗಮಿಸಬೇಕಾಗಿ ವಿನಂತಿಸ್ತೀನಿ. ಪ್ರೊಫೆಸರ್ ಶೇಖರ್ ಶೆಟ್ಟಿ ಅವರು ಕೂಡ ದಯಮಾಡಿ ವೇದಿಕೆಗೆ ಆಗಮಿಸ್ತಾರೆ. Thank you, sir, for your thought-provoking lecture. On behalf of the organizers, I now request our Honorable Vice Chancellor to felicitate Professor Appara Podale, the chief guest of today's program. Dear esteemed members of the audience, we have amongst with us Professor N.K. Loknath. He is a renowned physicist and material scientist who has made significant contributions to the field through his research, teaching and administrative roles. His profound insights have propelled the boundaries of knowledge, inspiring countless individuals in their academic pursuits. Currently serving as the 26th Vice Chancellor of the prestigious University of Mysore, Professor Loknath's journey has come full circle as he leads his own alma mater to new heights of excellence. Today we are privileged to have Professor N.K. Loknath grace us with his wisdom as he delivers his presidential address. Without further ado, I humbly request our Honorable Vice Chancellor to address this august gathering.
ಎಲ್ಲರಿಗೂ ನನ್ನ ನಮಸ್ಕಾರಗಳು. ವೆರಿ ಗುಡ್ ಆಫ್ಟರ್ನೂನ್. ಪ್ರೊಫೆಸರ್ ಎಸ್.ಆರ್. ನಿರಂಜನ, Vice Chairman, Karnataka State Higher Education Council, Government of Karnataka, Chief Guest of today's function, and an eminent resource person and former Vice Chancellor of Hyderabad Central University, Professor Appara Podale, and our senior distinguished Professor H.Shekar Shetty, and the Registrar, Mrs. Shailaja, and the Professor K.N. Amrutesh, Director IQAC, and this organizing this lecture, endowment lecture, and all the dignitaries of the dais and faculty members of various departments, students and media persons. I am extremely very happy today to be a part of this endowment lecture to mark the significant contribution of Professor H.Shekar Shetty, an institutional builder. At the outset, on the behalf of the university, I must thank Messrs. Pioneer Hybrid Seed Company, Private Limited, Hyderabad, for having instituted this endowment at the university. This seed company has recognized the services of Prof.H.Shekar Shetty in the area of seed science and technology, especially his research work revolving around and directly benefiting the various seed companies like plant diseases diagnosis, standardization of protocols for innate solutions to various diseases, diagnosis problems. At this juncture, I would like to inform that there are 15 endowment lecture series are here in our university, 30 chairs, 540 gold medals, and 50 cash prizes have been established at the university by various donors in honor personalities who have excelled in various faculties including science and technology, arts, commerce, education, and law. Such endowment lectures are highly beneficial for two reasons. One is to provide current and contemporary knowledge of research and development to the student community in particular and faculty by inviting professors of eminence and scientists, writers and policymakers and more. And also the second one is the endowment of this type of endowment lectures are really the assets of the universities which are always useful in establishing the collaborations between various institutions, universities, leading to exchange programs, and thereby we can strengthen our research and development activities in the university. Professor S.R. Niranjana, my good friend, has inaugurated this endowment lecture program with great pleasure and spoke about the important policy matters, especially he has guided the students how to become a good researchers of 14%. Sir, I would say that in India we are in the verge of very good higher education because there will be two factors are here. One is quality and one is the quantity. So in my idea and my physics principles is that two things cannot occur at the same time. If you go for quality, the quantity is the problem. If you go for quantity, quality is the problem. However, we have a very good number of students in our universities. We look forward for to be in the research in that 14% category. And he has also discussed about many things about the department, as well as he's always guiding the universities in many matters. After he took over as the vice chairman of KSHTC, he has organized various programs, and also he is very helpful to all universities vice chancellor to make the administration so simple, and has instituted so many innovative initiatives in the KSHTC. I request you, sir, one more thing. The universities are like giving an order from the state government that you should do this, you should do this. That means our autonomy is completely gone now. So there is even today, the admissions they will make the date. Which date only examination, which date only examination, this day only admission, this day only holidays. That should be taken up, sir. So I earnestly request you, sir, please look into the matter. Give us some autonomy, at least for our academic endurance, so that we can able to do better than this. And then you coming to the special lecture on innate immunity in plants with kite oligosaccharides delivered by Professor Padilla. It was truly a thought-provoking, very simple and thought-provoking and excellent speech, and I hope all the students will understand the importance of the immunity system. So I do not want to give more thing about Professor Apparao because he has been very expertise in this field. Though I am coming from the background of physics, I still understand how this innate systems as well as the oligosaccharides importance in the immunity development. And moreover, in Karnataka, Professor Dr. Timkapur was here. I am coming from the Malnad region. We have a very big problem of areca diseases. They are spending a lot of crores of money for this to solve these issues, but still it's under big, it is not under control yet. So I hope the researchers will look into this disease and take it this to be very serious because it depends upon most of the farmers in South Karnataka. And Professor Apparao explained in detail about innate immunity with suitable examples by taking his own research problems, opined that many plant-associated microbes are pathogens that impair plant growth and reproduction. And then the first branch recognizes the response to the molecules common to many classes of microbes, including non-pathogens. The second response to pathogen is virulence factors, either directly or through their effects on host targets. So I hope all the students, I hope nobody has taken the notes, I guess. Whenever you come out for the lecture, you should come up with a book, a small note, such that at least you could note down some references such that you can read. So that you can read better, you can understand better in future. And today we are all together here because of the research work carried out by the Prof. Shekar Shetty, the main attraction of this today's functions. He deserves this recognition to be a role model to many professors in the universities. I would say that I am seeing Professor, when I was a student of master's degree in physics, at that time the only laboratory of international standard is the Department of Applied Botany. Now it has been named as Department of Biotechnology. So that is his highly credentials to this. He encourages numerous students. I have seen many students, and also even though I am not a biology student, I know when I am in hostel about the donor media programs. So I am remembering many his students are working with us. So today that's very happy. So and he has resulted a very good successful career in his research as well as a national level, in the international level. So definitely the project initiated by him continue to have a significant societal relevance. His work on plant decision management and diagnosis are set in model for every researcher. I wish him a very happy, prosperous and healthy life and look forward his continuous guidance whenever university needs his expertise. As a Vice Chancellor, I am always strengthening the academic methods or academic teaching or maybe research. So therefore, I asked the IQAC to initiate this all type of lectures, including endowment lectures, should pass through IQAC. Such a way that the IQAC should have interact with all the departments. They can do all these type of lectures so that it will be benefited to the students and as well as the faculties of the universities. And this type of lectures would be helpful for all the people, those who want to pursue higher education more and more. As Professor Niranjana sir said that the higher education is growing and most of the students are studying in higher education in India. So you are all in Manasa Gangotri means you are very happy that you should be placed somewhere else because of your education. So higher education is very crucial in your life. I hope you will carry your higher education such that you can able to do something better by yourself, then to your family, then to the society. And finally, I thank the organizers for this endowment lecture program. Hope this type of lecture program will continue in our university, and all the best. Thank you, one and all. Thank you, sir.
ಆನರಬಲ್ ವೈಸ್ ಚಾನ್ಸಲರ್ ಭಾಳ ಸಂಕ್ಷಿಪ್ತವಾಗಿ ಏನು ನಡೀತು ಇವತ್ತಿನ ಪ್ರೊಸೀಡಿಂಗ್ಸ್ನ ಭಾಳ ಚೆನ್ನಾಗಿ ಮಂದಟ್ ಮಾಡಿಕೊಟ್ಟಿದ್ದಾರೆ ತಮ್ಮೆಲ್ಲರಿಗೂ. ಮತ್ತು ವಿಶೇಷವಾಗಿ ಡಾಕ್ಟರ್ ಪೊಡಿಲಿ ಅವರ ಲೆಕ್ಚರ್ ಯಾವ ರೀತಿ ಸಿಂಪ್ಲಿಫೈ ಆಗಿ ಮಾಡಿದ್ದಾರೆ ಅನ್ನೋದೊಂದು ಭಾಳ ಪ್ರಮುಖವಾಗಿ, ಮತ್ತು ಎಂಡೋಮೆಂಟ್ ಲೆಕ್ಚರ್ ವಿಚಾರವಾಗಿ ಭಾಳ ಸ್ಪಷ್ಟತೆಯನ್ನು IQACಗೆ ಜವಾಬ್ದಾರಿ ನೀಡಿರ್ತಕ್ಕಂಥದ್ದು. ಎಲ್ಲ ಈಗಾಗಲೇ ಎಷ್ಟು ಎಂಡೋಮೆಂಟ್ ಲೆಕ್ಚರ್ ಇದೆ ಅನ್ನೋದನ್ನ ತಮ್ಮ ಮುಂದೆ ಪ್ರಸ್ತುತಪಡಿಸಿದ್ದಾರೆ. ಎಲ್ಲರಿಗೂ ಅನುಕೂಲ ಆಗುವಂಥ ಕೆಲಸವನ್ನು ಈಗಾಗಲೇ ಭರವಸೆ ನೀಡಿದ್ದಾರೆ. ಇದೇ ಒಂದು ಸಂದರ್ಭದಲ್ಲಿ ನಮ್ಮ ಜೊತೆ ಕೈ ಜೋಡಿಸ್ತಾ ಇರ್ತಕ್ಕಂಥ ನಮ್ಮ ಡೆಪ್ಟಿ ಡೈರೆಕ್ಟರ್ ಆದಂಥ ಡಾಕ್ಟರ್ ಜೆ ಲೋಹಿತ್ ಅವರು ಮಾಸ್ಟರ್ ಆಫ್ ಸೆರೆಮನಿ ಆಗಿ 300ನೇ ಅಂಕಣವನ್ನು ಇವತ್ತು ಕಂಪ್ಲೀಟ್ ಮಾಡ್ತಾ ಇದ್ದಾರೆ. ಈಸ್ ಎನ್ ಅಸೆಟ್ ಟು ದಿ ಯೂನಿವರ್ಸಿಟಿ ಆಫ್ ಮೈಸೂರು. ವೇರೆವರ್ ಈಸ್ ದೇರ್, ಹಿ ಈಸ್ ಆಲ್ವೇಸ್ ವೆರಿ ಆಕ್ಟಿವ್. ಐ ರಿಕ್ವೆಸ್ಟ್ ಆನರಬಲ್ ವೈಸ್ ಚಾನ್ಸಲರ್ ಟು ಫೆಲಿಸಿಟೇಟ್ ಹಿಮ್ ಆನ್ ದಿಸ್ ಅಕೇಶನ್. He is also the coordinator of this program. Any endowment lecture for that matter, IQAC program, he is very effectively engaging. Thank you.
to extend heartfelt gratitude to all those who have contributed in making today's endowment lecture a resounding success. Now, I have the honor to invite a senior academician, Professor Ines Raju, who also serves as the chairman of the Department of Environmental Science, to propose vote of thanks.
ಸೊ ವೈಸ್ ಚಾನ್ಸಲರ್ ಮಧ್ಯದಲ್ಲಿ ಹೇಳಿದ್ರಲ್ಲ ನಿಮಗೆ ನಮಗೆಲ್ಲ ಸ್ವಲ್ಪ ಅಟಾನಮಿ ಕೊಡಿ ಅಂತ. ಅಟಾನಮಿ ಕೊಟ್ಟಿದ್ವಿ ಹಿಂದೆ ಭಾಳ, ಹಾಗಾಗಿ ಹಿಂದೆ ಅಟಾನಮಿ ಜಾಸ್ತಿ ಆಗ್ಬಿಟ್ಟಿದೆ. ಹಾಗಾಗಿ ಇವಾಗ ನಾವು ಏನು ಮಾಡ್ತೀವಿ ಅವೆಲ್ಲ ಪಾಲಿಸಿ ವೈಸ್ ಚಾನ್ಸಲರ್ ಮಾಡ್ತಾರೆ. ಅವ್ರ ಮೇಲೆ ಯಾವುದೇ ಥರದ ಒತ್ತಡ ನೀವು ಪರವಾಗಿಲ್ಲ. ಅಕಾಡೆಮಿಕ್ ಕ್ಯಾಲೆಂಡರ್ ಕೊಟ್ಟಿದ್ದೇವೆ, ಅದ್ರ ಪ್ರಕಾರ ನಡೀಬೇಕು. ನಿನ್ನೆ UGC ಲೆಟರ್ ಬಂದಿದೆ, ರಿಸಲ್ಟ್ ಎಲ್ಲ ಡಿಲೇ ಆಗಿ ಮಕ್ಕಳು ಬೇರೆ ಬೇರೆ ಎಕ್ಸಾಮಿನೇಷನಿಂದ ವಂಚಿತರಾಗ್ತಾ ಇದ್ದಾರೆ. ಆದ್ದರಿಂದ ಅಕಾಡೆಮಿಕ್ ಕ್ಯಾಲೆಂಡರ್ ಪ್ರಕಾರನೇ ನಡೆದು ಅದರ ಪ್ರಕಾರ ನಡೆಸಿ ಅದರ ರಿಸಲ್ಟನ್ನು ಕೊಡಬೇಕು ಅನ್ನೋದನ್ನು ನಾವು ಭಾಳ ಕಠಿಣವಾಗಿ ಅವರಿಗೆ ಅವ್ರ ಮೇಲೆ ಹೇಳಿದ್ದೀವಿ. ಫೀಸ್ ಸ್ಟ್ರಕ್ಚರನ್ನು ಸಹ ನಾವು ಅವ್ರಿಗೆ ಇದೇ ಥರನೇ ಫೀಸ್ ಸ್ಟ್ರಕ್ಚರ್ ಕಲೆಕ್ಟ್ ಮಾಡಿ ಎಲ್ಲರೂ ಹೈಯರ್ ಎಜುಕೇಷನ್ ಕೌನ್ಸಿಲ್ ಗೌರ್ಮೆಂಟ್ ಆಫ್ ಕರ್ನಾಟಕದಿಂದ ಪಾಲಿಸಿಗಳಾಗ್ತದೆ. ವೈಸ್ ಚಾನ್ಸಲರ್ ಅಟಾನಮಿ ಕಡಿಮೆ ಆಗಿದೆ ಅನ್ನೋದು ನನಗೂ ಗೊತ್ತು. ಅದು ಇರೆಗ್ಯುಲರ್ ಆಗಿದ್ದರಿಂದ ಅವ್ರ ಅಟಾನಮಿ ಕಡಿಮೆ ಆಗಿದೆ. ಒಂದೊಂದು ಯೂನಿವರ್ಸಿಟಿ ಒಂದೊಂದು ಟೈಮಲ್ಲಿ ಪರೀಕ್ಷೆ ಮಾಡೋದು, ಒಂದೊಂದು ಯೂನಿವರ್ಸಿಟಿ ಒಂದೊಂದು ಟೈಮಲ್ಲಿ ರಿಸಲ್ಟ್ ಕೊಡೋದು, ಒಂದೊಂದು ಟೈಮಲ್ಲಿ ಅಡ್ಮಿಷನ್ ಮಾಡೋದು, ಪಾಠ ಪ್ರವಚನ ಮುಗಿಲಿಕ್ಕಿಲ್ಲ. ಅದಕ್ಕೋಸ್ಕರ ಕಾಮನ್ ಕ್ಯಾಲೆಂಡರ್ ಮಾಡಿದ್ದೇವೆ. ಫೀಸ್ ಸ್ಟ್ರಕ್ಚರ್ ಅವ್ರಿಗೆ ಅಟಾನಮಿ ಕೊಟ್ಟಿದ್ದೇವೆ. ನಮ್ಮ ಟೈಮ್ ಟೇಬಲ್ ಪ್ರಕಾರ ಕೋರ್ಸ್ಗಳು ನಡೀಬೇಕು. ಆ ಟೈಮಲ್ಲಿ ಅವು ಕ್ಲೋಸ್ ಆಗಬೇಕು, ಇಂಥ ಟೈಮ್ಗೆ ರಿಸಲ್ಟ್ ಅನೌನ್ಸ್ ಆಗಬೇಕು ಅಂತ ಗೊತ್ತಿದೆ. ನೀವು ಯಾವ ಕಾರಣದಿಂದ ವೈಸ್ ಚಾನ್ಸಲರ್ ಸ್ಟ್ರೈಕ್ ಮಾಡೋದಾಗಲಿ ಕೂಗಾಡೋದಾಗಲಿ ಮಾಡುವಂಥ ಪರಿಸ್ಥಿತಿ ಇಲ್ಲ. ಅದೆಲ್ಲ ಕೌನ್ಸಿಲ್ ಮಾಡ್ತದೆ ಗಮನದಲ್ಲಿ ಇಟ್ಕೊಂಡು. UGC ಗೈಡ್ಲೈನ್ಸ್ ಅನ್ನ ಗಮನದಲ್ಲಿ ಇಟ್ಕೊಂಡು ಆ ಮಾನದಂಡಗಳನ್ನ ಉಪಯೋಗಿಸಿ ನಾವು ಟೈಮ್ ಫ್ರೇಮ್ ಅನ್ನು ಮಾಡ್ತೀವಿ. UGC ಬಿಟ್ಟು ನಾವು ಮಾಡೋದಿಲ್ಲ. ನಮಗೂ ಯಾರಿಗೂ ತೊಂದರೆ ಕೊಡಬೇಕು ಅನ್ನೋದು ನಮ್ಮದು ಉದ್ದೇಶ ಅಲ್ಲ. ಬಟ್ UGC ಹೇಳ್ತಿದೆ ನಮ್ಗೆ ಇಂತಿಂಥ ಟೈಮಲ್ಲಿ ಇಂಥದ್ದು ಆಗಬೇಕು. ಇವಾಗ ಟೈಮ್ ಟೇಬಲ್ ಸುಪ್ರೀಂ ಕೋರ್ಟ್ ಜಡ್ಜ್ಮೆಂಟ್ ಆಗಿದೆ. ಸುಪ್ರೀಂ ಕೋರ್ಟ್ ಜಡ್ಜ್ಮೆಂಟ್ ಪ್ರಕಾರ ಟೈಮ್ ಟೇಬಲ್ ನಡೆಸಬೇಕು ಅಂತಕ್ಕಂಥದ್ದು ಇಟ್ಸ್ ಎ ಜುಡಿಷಿಯರಿ. ಹಿಂದಿನ ವರ್ಷ ಕರೋನಾ ಬಂದಿದ್ದರಿಂದ ಸ್ವಲ್ಪ ಡಿಸ್ಟರ್ಬ್ ಆಗಿದೆ ಬಿಟ್ರೆ, ಕರೋನಾ ಬಂದಿದೆ ಅನ್ನೋ ಡಿಸ್ಟರ್ಬ್ ಆಗಿರೋದು ಬಿಟ್ರೆ, ಮತ್ತೆ ಈಗ ನಾವು ಕರೋನಾ ಅಂತ ಹೇಳ್ತಾ ಈಗಾಗಲೇ ಅರ್ಧ ವರ್ಷ ನಾವು ಮುಂದೆ ಇದ್ವಿ. ಹಿಂದಕ್ಕೆ ಬರ್ಬೇಕಲ್ಲ ಅಕಾಡೆಮಿಕ್ ಕ್ಯಾಲೆಂಡರ್. ಹಾಗಾಗಿ ನಿಮ್ಗೆ ರಜಾ ಎಲ್ಲ ಕಟ್ ಮಾಡ್ಲಿಕ್ಕೆ ಹೇಳ್ಬಿಟ್ಟಿದ್ವಿ. ರಜಾ ಇಲ್ಲ, ಏನಿಲ್ಲ, ಸುಮ್ಮನೆ ಓದೋದು ಬರೆಯೋದು, ಡಿಗ್ರಿ ತಗೊಂಡು ಮನೆ ಹೋಗೋದು. ಸೊ ಅದಕ್ಕೆ ಈ ರಜೆಗಳೆಲ್ಲ ಮಾಸ್ಟರ್ ಡಿಗ್ರಿನಲ್ಲಿ ರಜೆ ಉಪಯೋಗಿಸ್ಬೇಡಿ. ಇಟ್ ಇಸ್ ವೇಸ್ಟ್ ಆಫ್ ಯುವರ್ ಟೈಮ್. ಸರ್ಚ್ ಫಾರ್ ಯುವರ್ ಸೆಟಲ್ಮೆಂಟ್. ಅವ್ರು ರಜೆ ಕೇಳೋರು BSc BA ಓದೋರು, PUC ಓದೋರು, SLC ಮಕ್ಕಳು, ಕಿಂಡರ್ ಗಾರ್ಡನ್ ಮಕ್ಕಳು, ಅವರೆಲ್ಲ ಕೇಳ್ತಾರೆ ರಜಾ. ನಿಮಗೆ ಬೇಕಾಗಿಲ್ಲ. ಯು ಕೀಪ್ ಯುವರ್ ಸೆಲ್ಫ್ ಬಿಸಿ. ದಟ್ಸ್ ವಾಟ್ ಅಕಾಡೆಮಿಕ್ ಕ್ಯಾಲೆಂಡರ್ ಇಸ್ ವೆರಿ ಟೈಟ್. ದರ್ ಇಸ್ ನೋ ಪಾಯಿಂಟ್ ಇನ್ ರಿಲ್ಯಾಕ್ಸಿಂಗ್. ದಟ್ ವಿ ಆರ್ ಡಿಸೈಡೆಡ್. ಯಾರು ವೈಸ್ ಚಾನ್ಸಲರ್ ಗಲಾಟೆ ಮಾಡ್ತಾರೆ ಆಯ್ತಾ?
Very good afternoon to everybody. I am here to propose vote of thanks, and it is my privilege to propose vote of thanks in this special occasion. First of all, on behalf of University of Mysore, University Endowment Lectures Program, I sincerely thank our Honorable Vice Chancellor, Professor N.K. Loknath sir, for attending this function and presiding over this function. Since morning, he is with us to grace this occasion. Sir, on behalf of the University of Endowment Committee, I thank you profusely. And also I thank another chief guest today, our Vice Chairman, Professor S.R. Niranjana, Karnataka Higher Education Council, for attending this function, sir. On behalf of the university, I would like to thank you. And also I thank the Professor Appura Padale, who has given the excellent lecture on the occasion of Prof.H.Shekar Shetty Endowment lecture. So on behalf of the university and endowment lecture program, so I profusely thank Professor Padale sir. I also thank Prof.H.Shekar Shetty for attending this function, for whom name is the endowment lecture has been established. I thank you for attending this function. I also thank our registrar who attended this function and left the function in between because of her another program. On behalf of the University of Mysore, I also thank our registrar, Professor Shanija Madam. I also for all organizing committee members
who has attended this function, special program. Professor K. Amrutesh, who is the backbone of this function and organized this function very well. On behalf of the University of Mysore, I also thank Professor K. Amrutesh and IQAC Director and Chairman of the Botany Department. And also I thank all the members, professors from various departments, students from other departments and media persons and organizing committee for attending this function and make this function very successful. So with this, I thank one and all once again. Thank you very much.
ಪ್ರೊಫೆಸರ್ ಎನ್ ಎಸ್ ರಾಜು ಅವ್ರಿಗೂ ಕೂಡ ವಂದನೆಗಳನ್ನ ಸಲ್ಲಿಸ್ತೀನಿ. ಈಗ ನಮ್ಮ ರಾಷ್ಟ್ರಗೀತೆಯ ಧ್ವನಿಮುದ್ರಿಕೆಯನ್ನು ತಮ್ಮ ಮುಂದೆ ಬಿತ್ತರಿಸಲಾಗುವುದು. ರಾಷ್ಟ್ರಗೀತೆಗೆ ಗೌರವ ಸೂಚಕವಾಗಿ ದಯಮಾಡಿ ಎಲ್ಲರೂ ಎದ್ದು ನಿಲ್ಲಬೇಕಾಗ್ತದೆ.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे.
ಬಂಧುಗಳೇ, ತಮ್ಮೆಲ್ಲರಿಗೂ ಭೋಜನದ ವ್ಯವಸ್ಥೆಯನ್ನು ಮಾಡಲಾಗಿದೆ. ತಾವೆಲ್ಲರೂ ಭೋಜನ ಸ್ವೀಕರಿಸಿಯೇ ತೆರಳಬೇಕಾಗಿ ವಿನಂತಿಸ್ತೀನಿ.